Hello and welcome to the Xenothesis podcast. This is our third special episode where we'll discuss aging and immortality. And I'm joined, as always, with my co-host.
Richard Axon.
Hi,
Michael.
Richard.
How's it going?
Good, thank you. Yeah, this is our third special episode. First one was Cyberpunk. The time I remember what it was when Cyberpunk was just coming out, and we decided to do some Cyberpunk related, which actually relates to this topic in fact later. At some point.
It's a lot of them have kind of transhumanist-y themes, right? That's been a general pattern, and I went off on like medical device implants and the potential problems with doing that poorly.
And then we had the second episode was Gattaca, the movie, which also had very similar concepts, like more of developing a perfect human by genetically modifying the embryos type of thing, which was also in a way relates to this.
Perhaps a more historical view there, right? We look back over the history of the rather dark history of eugenics, and hopefully a slightly lighter future for genetic modifications and humans going forward.
Well, and fingers crossed.
Yeah, hopefully. We don't know.
We really don't.
The potential of the technology, right? If applied in a good sense, and then a bunch of as usual, a little bit of media analysis from us.
Yeah,
like to mix that in.
Yeah, absolutely. So today's episode, we decided to talk about biological immortality and aging, and the reason why we thought that would be a good idea. One reason is because of Richard's background, his research in aging biology. Would give us, a great opportunity to discuss this. And I think, also we both think this is a pretty exciting and interesting topic to discuss, because it also relates to a lot of what we've been doing so far. Our society, our world is developing technology that is making biological mortality and ceasing of aging more and more realistic day by day. And the fact that we in our Xenogenesis read through, we also see a lot of modified humans who can live longer. So it's a perfect opportunity for us to discuss these topics, to sort of put shine some light on them. Don't you think, Richard?
Yeah, absolutely. Yeah, I mean, so to open a little bit with the prospect of stopping aging, I still think is a little bit distant, right? But the very beginnings of that process, wherein we actually begin to delay it, we begin to hold it off. To a degree, we've already been doing that, right? When you look historically over the trends in lifespan, average lifespan in humans, then it's been ticking up massively over the decades. Although the maximum lifespan has still been a bit limited, right? I think the record holder is Jean Calment at 123.
Oh God!
She's held that record for decades now, so we haven't pushed it out. Haven't pushed it out much.
I would say that's more of an outlier. I would say, because on average, you should say it's around. Well, it depends on all the factors involved in that, but you would say that in a society where there's no conflict as such and no pandemics, I would say the average life expectancy would be maybe on average 78 years old with a good diet.
I think it's around in the UK. I think it's around about 80 now, maybe a little bit less. I think it's certainly for women. I think it's hit 80.
Yeah,
I'm fairly sure. Might be close to late 70s. But that transition's been that's happening fast around the world.
Yes,
because the risk profile for dying through life, most of the average gains we got in lifespan were from infant mortality going away because of our control of infectious diseases, and then our kind of reduction
general hygiene, introduction of hygiene and
hygiene and just safety standards, right? Yeah. You're a lot less likely to be killed by all kinds of random accidents, from falling off things to having things fall on you and crashing into all the safety standards in our societies have improved. But yeah, no, vaccination and infectious disease control and just understanding all that stuff means that the developing world is coming into that kind of lifespan transition way faster than we did when we first made that transition in the Industrial Revolution and so on, right? That like everyone else, as the world is beginning to come out of more extreme poverty in various places, right? You don't have to get as far up the sort of developmental ladder as slowly, right? The huge parts of the world are now living much longer than you think, right? Global average life expectancy is I can't remember what the numbers are. I should probably look that one up, but it's higher than most people would guess it to be. I remember that much. So that's been a huge improvement over time.
Yeah,
and it shows no signs of slowing down as of yet, though it has often been predicted that the extension of average lifespan will slow eventually. Lots of people argue that there will be a plateau at some point.
I to be honest, I would personally expect that would be the case, right? There's so much that our bodies can withstand, or the time, how much time they can withstand. But it's an interesting, it's a pretty amazing feat that the fact that introduction of, as you said, safety procedures and as well as general hygiene reduced the infant mortality, which basically showed that increased the population number and the life expectancy.
The amazing thing about it is we weren't trying to do that explicitly.
Yeah,
we were solving all these other problems indirectly, and now aging has actually kind of become one of the linchpin problems, right?
Yeah.
So we picked all this low-hanging fruit and we got a lot of additional life because of it. But now aging is starting to become the main problem, right?
Yes.
So when you if you try and target heart disease or dementia or cancer, you don't actually gain that much in expected lifespan by curing any one of those. Right.
Yeah.
So if you cure all cancer completely, you get 3 extra years or something, because there's something else that will kill you, like heart disease or Alzheimer's or one of the other systems that will have a problem with it. So it's now the case that just the process of us aging is kind of one of the main bottlenecks in addressing the sort of long-term chronic conditions that are now the bulk of the problems that the medical establishment has to face when we're not dealing with pandemic infections.
Yes, absolutely. Before we continue, let's maybe clarify certain concepts that we will talk about today. Especially, we mentioned a few things. Aging. We should probably explain what do we mean by that. And here,
let's do some defining our terms.
So, aging wise, this is from Encyclopedia Britannica. Aging is the sequential or progressive change in an organism that leads to an increased risk of debility, disease, and death, eventually death. Senescence consists of these manifestations of the aging process. And so that's basically the definition. We'll explain more of some of the aspects of what I just read in a second. But let me first of all clarify what we mean by immortality. And here, today we will talk about biological immortality, meaning where the process of aging has been stopped, right? It doesn't mean that we don't die, right? It's still possible for our bodies to die from many different aspects, accidents, everything. We're talking about the fact that our bodies wouldn't change with time. That type of immortality.
No, actually, I'm not sure that that's necessarily an entirely coherent definition of immortality, even in the biological sense, because the idea that we don't change with time, I think, is actually going to be antithetical to being properly immortal, right? I think change is going to be inherently necessary in some minor degree for that to be the case.
But I think that's the reason why we have to, today, we will clarify those points, because we have a lot of things to talk about. So those things that we'll mention about later on I think will really clarify what exactly we mean by that. Because honestly, looking for a definition of immortality, like obviously you can find the true immortality definitions by biological immortality. It's not really well defined because there's so many factors affecting it that it's quite hard to pinpoint.
Yeah. I think it's actually worth spending a bit of time here exploring this definition, because it's
Sure.
Let's for a second return to the aging definition before we tackle immortality. So one of the other ways you can think about aging is from a statistical perspective, right? So if you say like, what is my risk of dying now, right? I'm 30, so it's still like, well, not quite yet. I'm 29. I'm almost 30, so it's pretty low in absolute values, but it's raising over time, right? Yes. And it will increase almost exponentially. Not quite. Technically, it's logistic, but it increases almost exponentially with time. And there's kind of a bit of a dog leg in that graph at around about 60, 65, where it starts to get really bad, right? And once you're in your 90s, it's like a dice roll if you're going to make it to the next year. So it's
true.
Like your risk of dying increases substantially over time. So one of the ways you can think about aging or the cessation of aging is just having a constant risk of death, at least an intrinsic risk of death. Right? Let's ignore the being hit by a bus type causes, but like the probability that you will just keel over dead for biological reasons being constant across time, and that constancy would be considered like negligible senescence, right? That's to say, there is minimal aging, right?
Yep.
Or barely noticeable aging, which is sort of one of the that's not quite immortality in the like the literal living forever sense, or at least like literal living, say, as long as the universe will continue to exist sense, but because you can still have some fixed probability of dying, right? If you retained your 30-year-old's risk of dying indefinitely, you would still eventually die for biological reasons because of some weird low probability outlier event. So that's like we should kind of bear that in mind when we think about what it means to be negligibly senescent, right? It's not quite immortality. So that's kind of like one of the, I suppose, like medium-term goals, I suppose you might say, of like the anti-aging project as a whole would be to get to negligible senescence for humans, rather than have this exponentially increasing risk of death over time.
Absolutely.
And there are some examples of organisms in nature which kind of fit that pattern, which we will get onto. But then, so that's not aging per se, but then there's immortality. That's kind of a slightly different question, right? Because now we've got to start talking about what exactly is the timescale we're talking about immortality on, and what are the like upper bounds on what you might consider like possible immortality. So the universe, as far as we can currently determine, has a finite amount of negentropy, right? So we'll run out of energy to do stuff in the universe eventually, right? It will die a heat death in an extremely distant future. So that's like unless we figure out something about cosmology that lets us move to other universes, which who knows, then there's kind of a hard upper cap, right? So it's not like infinite, but it's like multi-billion year kind of upper end of what you might theoretically get to within the lifespan of the universe, as far as we know, based on current physics. So, somewhere between those two things of attaining lifespans that might be in the thousands of years, if you got to like true negligible senescence at a low constant risk of death, and you lived in a society that was even safer than ours is today, and then there's the much bigger numbers.
Yeah, but the thing is, with those numbers, like the statistics of like with the time passing, also the statistics of like the risk of death over time is obviously going to increase, because with the more time you're alive, it's basically a time, almost linear relationship. Basically, with the more time, the more risk happening. Like the chance of a bus hitting you, technically, over a million years, is going to be quite substantial.
And one of the sort of societal consequences we might get onto is what living longer might do to our risk tolerance.
Exactly. But you mentioned some of the examples of immortality in nature. There are some interesting concepts, but do you want to sort of finish off with the definition of the aging first before we go to this?
Let's. So, I think it's worth getting into this now, actually. So one of the distinctions that exists between an individual organism and a population of organisms is very interesting, right? Mm-hmm. Because life, in general, is basically immortal, right? Barring extrinsic threats that wipe out Earth and any other places where life is a thing, it keeps going forever.
Yep. In different forms, but basically
in different forms. But so the tree of life has a last universal common ancestor, right? We are all descended from an original organism, and we have a common individual type of organism, right? Yep. We have a last universal common ancestor, so our lineage is shared. All of the life on Earth has a family tree that traces all the way back to this original organism, and that poses an interesting distinction, right? Because that lineage is immortal, but we as individuals are mortal, and it's worth asking the question why. Like, what's the difference? This is a little bit paradoxical, and a lot of the people who a lot of the people who talk about aging will bring up the thermodynamic arguments about aging, right? Because there's a lot of what I think of as some degree of confusion about this point, because individual organisms are energetically open systems, right? So in theory, the second law wouldn't necessarily apply, right? Because you can put energy in, you can make them last longer. But this actually misses one of the fundamental parts. Like, the other half of the picture that everyone misses when they talk about the second law is information, right? Information and entropy are just like two sides of the same coin. They're the distinction that separates individual organisms from populations of organisms is not energetic openness, but informational openness.
Yep.
Right. So we as individual organisms are energetically open, but informationally closed. Right. I'll try and explain what I mean by that by comparing us to populations of like individual organisms to populations of organisms, because the distinction, the thing that separates those two things, what provides the informational openness is evolution. Right. The process of evolution is what couples our environment to our genomes. Right, that's the information loop that needs closing. Right, you have to have a way of your environment shaping the genome, which is in many ways a piece of information that represents important information about your environment. Right, your genome is a snapshot of the environment in which you live, or a kind of mirror of the environment in which you live. Right, it provides a whole host of information, sort of bait into the assumptions that it makes about where the organism is going to be situated, what's going to be available to that organism, what's going to be around it, right? The genome is deeply reflective of all kinds of nuances about what's going on in your environment, and how it gets that way is natural selection, right? So as you have as you have all of these individuals in a population, through a process of non-random death, right? Some of them die off, and in a sense, the individuals in a population that die off we're sort of deleting bad information, right? Their genomes were making poor assumptions or not accurately reflecting some component of the picture of the environment that they would have benefited from if they had, which meant that they would have stayed alive, right? Statistically, right? Obviously,
no, absolutely.
There's a lot of errors in there.
Yes, as you said, excluding the random deaths. That's basically what natural selection is.
Yeah. So that shapes the genome of a population, right? That closes that loop. And it means that the germline, right, the reproductive cells, that lineage that traces all the way back to Luca, last universal common ancestor, that is under selection, and that has information that reflects the environment that is constantly subject to update. But any given individual does not have this feedback loop, right? We as an individual are not
that's
evolving.
Exact, yes.
So our genome is kind of an instantaneous snapshot of that ongoing process, right? It's a slice in time where a slightly imperfect representation of the environment that can no longer be updated within us. Right? There is no mechanism by which our environments can alter our genome to better reflect how that genome should function in that environment. Right? We just have a snapshot that we're stuck with. So one of the pieces of terminology I find useful to think about in this frame is Nassim Taleb's concept of the distinction between robustness and antifragility.
Okay.
Right, so you have fragile systems which are sort of, they break, right? And then you have robust systems which can maintain state. And then you have antifragile systems which get stronger under stress. Right? So the analogy there is, you can think of it in some mythological terms, right? Your fragile state is the sword of Damocles, right? A sword hanging over something precious by a single hair. It's fragile. It's easily broken. And then your robust systems are a phoenix, right? They maintain state, right? You can kill them off, but they'll come back the same way.
Exactly.
And then your anti-fragile systems are like hydras, right? You cut off a head, you get two more, it just gets stronger.
Yeah.
And as individuals, we're pretty robust, but we're not anti-fragile with respect to our genomes, right? The stresses of our environment cannot improve our genomes. But evolving systems, populations of individuals, stresses to their environment can improve their genomes, so they're anti-fragile. Right? So the process by which we as individuals can become immortal, not merely unaging, is to transition from being merely robust to being anti-fragile with respect to our genetic information. Right? That's what you have to achieve to become actually meaningfully immortal. And that requires a feedback loop.
Yeah, basically it would mean that we would have to be able to respond to any environmental changes. Like, for example, giving what we've been doing in Xenogenesis trilogy, having Oankali responding to the environment changes by basically modifying their bodies to whatever was necessary for them, right? So in here as well,
exactly.
Our bodies would have to be able to mold themselves in any way we want, depending on the cues from the environment.
So this is a theme that showed up quite a lot in this series of books so far, and I've discussed it before, is this like fusion of genetic and memetic information. Right, or the ability of memetic information to feed back on genetic information, right? The capacity to see and edit and control your own genome so that you can make it better reflect the environment in which you now reside.
Yeah, which is, but then there are some, obviously now, obviously as you mentioned, the life itself is immortal, but like the branches that have been developed, that have developed over the last millions of years, have come up with a quite range of ways to sort of not answer what you're making the life individual anti-fragile. Although there are there is an example I think that we could sort of say in a way, but it's sort of like to pseudo answer that question, right? So in here, for example, I found an example where we basically in its name provides us an example of an animal, of an immortal jellyfish. Its Latin name Turritopsis dorini, and basically where a jellyfish, its life cycle makes it grow from an egg to an adult jellyfish, which it then can reverse the state back to its polyp. Right, so it basically reverses its back to its functional beginning to again restart the cycle, and in a way, the question then is, as you were saying, Richard, like, is that the feedback to the environment? Is that the way to respond to changes to environment that are necessary for the jellyfish to modify? Obviously, it's not. I don't know. Of course, I'm not sure. Like, there's any research done on it, but like, if the jellyfish changes its genome in any way to respond to its environment when it goes back to a polyp.
So the immortal jellyfish, this hydra thing that it comes up a lot in this conversation. Like the feature that gives it this property is kind of the lack of distinction between the germline and the soma in this organism. Right, and this is a common theme to lesser or greater degrees in some of the other things that are somewhat immortal, right? So let's just actually define those terms, right? So the germline is your reproductive cells, yes, right? Because evolution cares about reproduction. That's this thing, and that's the that immortal lineage that we've talked about, right? That distinction. That those are the cells that reproduce. Those are the cells that move into the next generation. And then the soma is the cells of your body, right? The cells that make up, your organs and your fingers and your hands, but they don't get directly, like their genetics is not in the lineage that goes to the next generation, whereas your germ cells are.
Yes.
So in a sense, the soma, and this is one of the aging theories, there are many frameworks for thinking about aging processes, is the disposable soma theory, or the disposable soma framework, right? So, evolution doesn't care about your body in anything other than like it's as a means to an end to your reproducing, right?
Yeah.
So
that makes sense, right? As long as as long as you reproduced. I mean, the meaning of life is as long as you reproduced and create an offspring to continue that the immortal life lineage, as you called it, the life doesn't care like what happens to you as an individual, right? It's as long as the continuation of that lineage.
Yeah. So your body is a disposable vessel for the capacity of your reproductive cells to get made into another body, right? Yeah. That's evolution has this sort of tautological, self-referential logic of, right, yes, those things that are best at surviving survive and get better at surviving in the process, right? It's just this little loop, right? So that to apply that again to this hydra, right? The if you take like a given cell from a hydra, it can basically just regrow another hydra.
Yes.
Right. So there's not a distinction between the cells that are reproductive and the cells that are soma. Right. So it kind of cares about all those cells, which means that there's not really, like, it's so small and uncomplicated that the line between the individual and the population is somewhat blurred.
Absolutely. Yes.
Right. It's not like there is an individual hydra that is reproducing. There are individual hydra cells that are themselves mortal, but the individual hydras are kind of like blurred with the population.
And I guess this is actually quite common for as you said, for simple organisms that basically rely to counter this sort of issue of aging, right? That's basically what the evolution came up with. The conclusion was like how to solve this sort of age and immortality concept, right? Where there's no individual as such. The individual, as you said, still doesn't count, but basically everything is the same. It's a copy of itself.
Yeah, so you see these patterns of kind of near immortal things where there is a blurring between the lines of individual and population. And you also see it quite a lot in plants, right? So I think like the oldest known tree is the bristlecone pine, Pinus longeva. I'm not sure how to pronounce that. Something like that. The long-lived pine, right? Literally,
basically,
I think. Yes. Yeah, right. Which is like 5,000 years old in California somewhere, which is like 5,000 years old is just nuts, right? That's like older than a lot of human civilization.
Yeah.
Like this tree has like basically all the modern Abrahamic religions. Like it's about the same age as like Judaism.
Basically, yes.
It's kind of nuts. So yeah, like all of human history, this tree's still been like sat somewhere in California. All of like recorded human history.
It's crazy, and the reason why it's so old is because it's just cloning, almost basically cloning itself, on and on and on again.
To some degree, I think
Yes.
is well, I mean, I'm not sure about the specifics of this bristlecone pine, but I think so. A lot of the other long-lived trees like yews and so on, the way that they grow is kind of like they just sort of modularly add bits to their individual, right? So they have a reproductive cycle, they do sexual reproduction, but a given individual is also just kind of modular, right? You know, you can do this thing with plants where you graft them and grow grafts clonally, right? So that whole ability to clonally reproduce blurs the line between individuals. And then you have like species of tree where basically the entire forest has an interconnected root system and they just put out suckers and grow up new trees. So that individual or population level thing is blurred in many of these long-lived plants because they have this modularity of developmental structure, unlike animals, which have a very fixed developmental pattern.
No, absolutely. I was going to mention the whole root system because I think that's how the bristlecone pine actually, for a lot of the forests that are involved, of the bristlecone pines are basically, all those trees are almost basically interconnected by the root systems, and basically, it's just basically one massive organism. That just keeps popping up more and more copies of itself.
Mm-hmm.
It's a really fantastic and interesting concept that we will discuss today in more sort of detail. And there are in biology, we mentioned few concepts today like senescence that we still have to define. But basically today our major conversation will involve talking about what are the hurdles to achieve biological immortality? What are the requirements really not requirements, but more like requirements and stepping stones in biology that we are yet to, that we have to address.
Yeah. And we're still mostly talking about kind of improving our robustness in those early definitions rather than necessarily transitioning to a state of antifragility.
Yes, absolutely. No, after Richard defined what exactly what the antifragility and robustness of the organism. And here we're still talking about robustness because, as mentioned earlier, we still lack the feedback loop, right? To reach that state of being able to respond to the changes to environment rapidly enough to but I guess let's start with some of the concept that we mentioned. Let's talk about senescence, because we mentioned senescence a few times here, and I think this requires a explanation what senescence is and why it's important for reaching biological immortality, why we need to reverse senescence. If I may, I will sort of explain what senescence is briefly. This is the definition basically of senescence, where it's a process in which a cell that accumulated too much damage or mutations within its DNA activates a system that it doesn't kill the cell, but which usually is, whenever something happens, a lot of cells just decided to undergo apoptosis, for example, which is a process of cell death. But in here, we don't do that. Instead, it just basically stops its usual replication cycle. So in biology, you have this cell cycle where there's a DNA condensation and then duplication, and then the cell splits, and then you have two identical cells, right? That's sort of this sort of cycle, and it repeats and repeats and repeats. The cells basically, they have a feedback system where in case something goes wrong, the cells have the capability of basically saying, my death is probably not necessarily, it could be energetically unfavorable. I might as well just stop doing what I'm doing and just, sit there and do nothing. And these cells become dormant with time, and with time, as as the time progresses, they accumulate in our bodies, right? So that's basically what the problem of senescence is that we have cells that are not doing anything. They probably accumulate too much damage, and to for the safety of the organism, they just decided not to basically stop doing what they were doing and just sit there, which is not ideal.
A couple of things from that. Firstly, they're not entirely doing nothing, right? They have this senescence-associated secretory phenotype, which is SASP, bit of a tongue twister. So they secrete, cytokines and other chemicals into the environment of the cell that, under normal circumstances, should recruit macrophages and other immune cells to come in and eat them.
Yes.
Right. So it's kind of the one of the interesting properties of the soma is that it's capable of a certain amount of selflessness in the evolutionary sense, right? Because the unit of reproduction is the individual, not the cell. So you have cell behaviors which appear altruistic despite the selfishness of the evolutionary process in organisms, because they're in the same boat, right? In order to get their genes into the next generation, theoretically they have to play a positive sum game with all the other cells in the body to do something positive, right? So that's where you can get these kind of like selfless behaviors of cells killing themselves when they think that there's something wrong with them that would negatively impact the organism. We're still kind of anthropomorphizing, but it's close enough.
Yeah.
But so one of the things that these senescent cells have kind of failed to do is take the apoptosis route, which is the normal cell suicide route. But instead, if they've maybe they've kind of lost confidence in their ability to do that well in some sense. So instead, they're recruiting immune cells to come along and kill them with this secretory phenotype. But that doesn't continue to work as effectively throughout life. So, as our immune system ages, the capacity to clear senescent cells decreases, and thereby their numbers increase. And then the inflammatory condition that they generate to recruit immune cells to kill them off, because they're perhaps concerned they might become cancerous. Again, a bit anthropomorphic, but fine. Like that no longer works, and now we have side effects because of all of this SASP stuff, which is causing inflammation and the accumulation of macrophages and overwork. The immune system's got a lot of stuff to do. It's got noisy signals now. So yeah, what is an okay mechanism for trying to prevent cancer in early life becomes a problem as you get older.
Yeah.
Which actually leads us into one of the other kind of useful conceptual frames to think about aging, which is this concept of antagonistic pleiotropy. It's a bit of a mouthful of a set of words, but it's like pleiotropy just means to have multiple functions. And then antagonistic is obviously like antagonists, right, pulling in opposite directions, as it were. So like a function can have or a biological thing can have multiple effects, right? So the senescent cells have a positive effect in that they try to prevent cancer, but then later on they have a negative effect in that they have this increased background inflammation level. But natural selection is concerned more about stuff which is happening earlier on in life, prior to you becoming reproductive and during your reproductive peak. More than it is concerned about stuff which is happening after that point. Right? This is a concept called the selection shadow. Right? So selection is less effective as you get older because it's having less defects that arise later on having less impact on your ability to reproduce. So this antagonistic pleiotropy thing comes along with, okay, it's beneficial to not get cancer when you're young, which means it gets selected for, but there's not that much selective pressure left to get rid of the negative side effects later on, right? So it has a positive near-term impact on your ability to reproduce and survive to reproductive age and raise kids and whatever, which is not dying of cancer. And then it has a negative effect later on, which is actually potentially increased risk of late-life cancer and immunocompromised to some degree, or less effective immune system, and other kind of general problematic side effects from all of those cytokines and stuff, right? That's no longer able to be selected against because it doesn't matter so much for your reproductive success if you die when you're 70 than if it does when you die when you're 30, right?
Yeah, absolutely. So in the reason why we mention why senescence is one of the concepts that need to be addressed in biology is that to reach biologic immortality, we need a reversal of that sort of those processes that lead to, well, first of all, prevention of the processes that lead to senescence, and also reversal of the senescence senescent cells of their senescent state. This is a concept that it's pretty interesting because it leads to other issues that are related to cells in general. Like, there's a lot of things that we can mention here. Should we just sort of list of things that could lead to sort of cell damage and stuff like that, Richard? And then we can pick something off and then start from there. What do you think?
Let's see. I mean, we could talk about like some of the descriptive frameworks for aging processes, like the hallmarks of aging.
Mm-hmm.
Yeah, that's probably a good place to start. I mean, there's a variety of different like descriptive frameworks for aging, and there's disagreement about which of them is good and which of them is bad. And as you might expect, in all such things, people think we should add categories, we should merge categories, we should remove categories, but the kind of the canonical set of hallmarks are you've got your primary hallmarks, which are like the causes of damage, and that's the genomic instability. So anything that's sort of damage to the genome, be that small or large scale mutations, and the mitochondrial genome, because as we discussed in one of our previous specials, the mitochondria has its own genome. There's telomere attrition. So if you've heard something about aging, you may well have heard something about telomeres. They got quite a lot of press. They're perhaps marginally less important in some sense than they were perhaps initially thought to be. But they're still they're still solving an important problem. They still have a lot of relevance to cancer biology, and there is still a kind of fundamental process that needs fixing, right? So telomeres are the regions at the end of our chromosomes, which they form like a protective cap at the end of our chromosome, and they exist because there is a problem in the way that our DNA replicates, so-called end replication problem, right? We have linear DNA, which means that right at the very end of the DNA, the machinery which normally replicates it needs a template, right? So when it's right at the end of the DNA, the other strand that it's going to use to build a new piece of DNA is not present, right? So it's like trying to build a brick wall whilst standing on a brick wall, right? It's difficult to add the bricks at the end while you're still standing on it, right? There's this fundamental problem of like, how do I put this here while standing on this thing, right? So there's not a straightforward solution to that problem. So what evolution has done to fix it is like copy paste a repetitive sequence right at the end a bunch of times, and then occasionally we have a special process which makes that longer with a telomerase enzyme and a little piece of extra template. Right, a little piece of template RNA that can go to the end of the genome and just paste on a few extra repeats to the end. But it does mean that eventually you would reach a crisis unless you had the telomerase enzyme turned on to keep making these longer, these repetitive regions that don't do anything other than solve the end replication problem. Eventually, you'd run out of telomere sequence, and you'd end up in important, useful stuff. Because the chromosomes will just get shorter over time. And I think these originally sort of came up in measuring like how many generations a cell could survive.
Yes.
Individually with it was Leonard Hayflick, and sort of measuring the loss of telomeres and considering that could be a mechanism by which you put a hard cap on how many generations of replication a cell could undergo if you have your telomerase turned off. But the kind of the flip side of the telomere situation is that if you turn telomerase on, you have the capacity to replicate indefinitely, which sounds a little familiar to some of the other things that we have a problem with, i.e. cancer, right?
Yeah.
So cancer replicates indefinitely, and most cancers, I think something in the ballpark of 70% have telomerase turned on, if I remember correctly.
This was pretty much a decade or so ago. Telomeres, the whole presence of telomeres was a big thing, where there was talked about like, oh yes, immortality to be achieved, the length of telomeres. And then when our understanding of cancer grew, we realized that actually a lot of cancer has, quite a substantial number of cancers utilizes the few telomeres to basically induce the continuous replication of cells, which is not exactly what we need in this point, right?
Yeah. That's. I mean, like you do want the ability to turn that on, but you don't want, because you need to be able to replace some stuff, but you don't want that to just grow out of hand, right?
Yeah.
So you need you need some control over that situation. Otherwise, like the uncontrolled version is cancer.
Exactly.
And like even the cancers that don't turn on telomerase have some alternative means of lengthening telomeres that likely involve some of the DNA replication the DNA repair mechanisms.
And this is, I think, a good point to actually mention, because earlier you mentioned DNA damage and DNA repair just now, and I think this is one of the other one of the biggest problem that I think, in my opinion, that is involved in that's a hurdle for biological immortality, is because with time, obviously, our repair system, our DNA repair, we have several checks in our cells to ensure that whenever DNA was replicated, it's sort of, there's a damage control, right? Because it's not always ideal, and there's things that can happen spontaneously even, right? But our the DNA repair systems, although very efficient, they're not perfect. And they're very good. Don't get me wrong. Obviously, if they weren't that very good, a lot of we wouldn't live as long as we do.
So one of the important things to think about for DNA repair systems in the context of the bigger picture of aging is there's an energy trade-off, right? In order to have a high fidelity of information, you have to spend energy to do that, right? That's just if you go back and look at Claude Shannon's work on energy and information theory in signaling, right? One of the ways you can think about aging is as an information transmission problem, right? You're trying to send a piece of state, right? Some description, some series of bits forward in time, and you want to do it reliably.
Yes.
But in order to send information reliably, you have to have mechanisms in place, like the checksums and Hamming codes and stuff that you can use to check that the information is actually correct based on what you sent forward, and trying to clean up noise in that signal, and it costs energy and additional bits of information to have redundancy and to maintain that, right? So effectively, as the reliability of the information transfer approaches perfection at 100%, zero chance of any errors, the energy you have to expend to maintain that degree of accuracy increases to infinity, right? So as you approach perfect information, you approach perfect energy expenditure to get there, right? That's asymptotic, right? So there's a finite degree of energy available, and you can spend it either on ensuring that the state is transmitted forward in time well, or on other things, right? So it's another one of those energetic trade-offs in the evolutionary process is I can spend money ensuring my genome's copied well and my epigenetic state continues to be what it should be. Or I can spend energy and resources on like quickly throwing together some additional eggs so I can reproduce right now.
Yeah, I mean, obviously, with energy expenditure wise, multiple, offsprings is more energetically beneficial than maintaining a body for a period of a long time.
It depends on your life history and your reproductive strategy, which is the best approach there, right? So it's the difference between R and K strategy reproduction, right? So you either go like big, high investment, very slow, long childhood, small number of individuals, high probability of success, or the other route, you just go, I want a gajillion offspring and like 99% of them are going to die, and the very few that make it, they're the lucky ones.
Yes.
Right?
Yes.
So in the lots of offspring route, then it can often be beneficial to do that early, to expend all of that reproductive energy on that early, although not necessarily always. So fish is sometimes a bit of an exception to that. Depends on the level of extrinsic threat from the environment. Right. So if you have a high extrinsic threat, if you have a high risk of predation or some other form of damage, then it makes a lot of sense to try and reproduce now, right? Get to adulthood and get kids as soon as possible, because there's a high probability something outside your control is just going to kill you. But if you have a low extrinsic threat, then it can make sense to wait and have a robust body, because then later on you may have a disproportionate chance of success. So, for example, like in fish, there are older females retain their reproductive capability for quite a long time. And fish grow almost continuously in a lot of species, right? So if you can get a big, bulky, older female that's still in peak reproductive success, right, she can generate a ridiculous number of offspring, right? Which means she has disproportionate ability to succeed in getting offspring into the next generation. So the selection shadow, right, is not extending very far in that context because of that reproductive strategy and because of the environment of degree of extrinsic threat. If she can get to an adult state, she has a low risk of dying, and therefore a high probability of success, disproportionate success, right? So you can get these situations where the trade-off between losing the ability to get to, or the potential to get to an older age and thus reach an area where you have lower extrinsic threat, because the big adult fish have low extrinsic threat, even if they have high extrinsic threat early in their life cycle. It can be worth the trade-off to expend the energy on the possibility of having that kind of a robust body to get to the adult state, which has differential success rates, right? So there's a lot of complexity in how the life cycle works out, what the threat model is, what the reproduction model is, that dictates what specific life course and lifespan evolution is going to select for.
No, absolutely. And just to finish off with the whole DNA damage, there are some sort of means in our bodies to increase the efficiency, maybe temporarily, of the DNA repair, the so-called longevity genes, which code for proteins that assist of the DNA repair. But all in all, as Richard said, this repair mechanism requires energy, and with the more perfect the repair would be, the more energy we need to input into that system. And as the conversation, as Richard explained, there's evolution chooses, in a way, picks which way it's the best for the given organism.
I mean, to talk a little bit more about the genomic instability stuff, DNA damage subject. So one of the reasons that we think that matters and is relevant for aging is many of the so-called progeriod diseases, right? The diseases which resemble accelerated aging. So kids who have these rare, often monogenic and inherited conditions where they appear to age very rapidly and die of age-related symptoms, heart failure, that kind of thing. And often dementia-like symptoms. Right? It depends a lot on the particular condition. But many of the single gene mutations that cause those progeriod conditions are in DNA repair things. The Bloom and Werner helicases, which are involved in DNA repair and quality control stuff for DNA replication, and a variety of other related systems. Right? XBA and XBC and for transcription coupled repair in xeroderma pigmentosum. Right? There's a bunch of these conditions which have a related age-related phenotype and then DNA damage related mutations. So that tells us that at the very least the DNA damage, if it gets bad enough, can be limiting. But if you look at the kind of rate of accumulation of DNA damage, if you take a look at the end of life, right? Take a whole bunch of different organisms with very different lifespans, very different body masses, and you say what's the mutations per unit of genome size when they die? That's roughly constant. Right? There's a similar amount of mutation when organisms die. In terms of mutation density, irrespective of how long you live.
Can I ask
or how big you are.
What are you basing that on? Is there any...
There's a recent Nature paper.
Oh, okay.
And that pattern is particularly interesting. It might tempt us to kind of take a causal picture, right? It would tempt us to say, okay, we know it revives one of the older hypotheses in the aging space, this kind of Orgel's error catastrophe, or related a bit to this concept of Muller's ratchet, right? Where you accumulate enough mutations where it's too many to deal with, right? It's a critical mass of mutations, and that's what kills you off. But that's not necessarily the case, right? It could well be that there are other systems in which these errors are accumulating, right? That it doesn't causally imply that because there are a similar number of mutations at the end of life, that's what caused the end of life. It just means that a similar amount of error tolerance across systems might be, there might be an error cascade, but it might not necessarily be purely DNA damage related. So just because we kind of started talking about the hallmarks and then we didn't quite pull that together. So we had the primary hallmarks, we were talking about genomic stability, we went back and forth about that a little bit, and then we did telomere attrition. And now we're getting into the kind of epigenetic space and a little bit into the proteostasis space, which are the next two of the primary cause of damage hallmarks.
Could you please explain to us, Richard? I mean, you've been doing most of the talk anyway here, being the expert, but could you please explain to us what epigenome is, so that we're all on the same page, and then let's go on from there.
Absolutely. Yeah, so that's the other layer of information I was talking about, right? So we've got this genetic layer of information, which is the sort of digitally encoded information in our genomes, right? It's the A, T, G's, and C's that you see on the double helix in all the movies, right? And then the next sort of layer of information on top of that are the annotations to the genome, if you will, are the epigenetic alterations, right? They are responsible for the developmental patterning that we have, right? Because every single one of our cells, all 40 odd trillion of them in an adult human, has the same genome, right? It's the same sort of recipe book from which we're working to build all of these different cells, right? We have brain cells and liver cells, and if you're lucky, they're doing different things.
But they all have the same thing. So how do we control?
Exactly.
What do they become? To what do they become?
So that's where the epigenetic information comes in, right? So on top of the genome, there's a couple of kind of layers of annotation, right? So your genome itself can be modified slightly. So you can have methylation marks where an extra little methyl group, 3 hydrogens and a carbon, stuck onto a cytosine. but it doesn't affect how the cytosine pairs. So the C will still pair with a T if you have the methylation mark. But in addition to that, all of your DNA, right, like every single one of those 40 trillion cells has like 2 meters of DNA in it. Which, that's like, I haven't done the multiplication, but like that's a really large like length, right? So it has to be all curled up in order to fit inside each cell, right? It's a single cell's like 30 micrometers at most, something in that order, and it's got to fit 2 meters of DNA in it, right? So it's all curled up very tightly, right? So these little disc-like proteins called histones, around which your DNA is wrapped. In these kind of little coils. So it's about 150 base pairs of DNA per coil, and then they all they supercoil, right? They stack up on top of one another in these little discs. So you have this kind of densely packed structure, and the histones themselves are host to a lot of the epigenetic information, right? They have little tails that protrude from where the DNA is wrapped around the disc, right? So the surfaces of the disc have some little bits that kind of come out the edges, these tails, which can carry annotations to the protein. So that they can be acetylated and methylated at lysines and arginines, and carry these additional marks, right? And then you have all these other proteins that kind of pay attention to the methylation state and to all
those changes in the histones and DNA.
All those pieces of information that are encoded on the histones, right? And what the histones do is kind of determine, or what the epigenetic information does is determine the accessibility of the piece of genetic information by the rest of the systems that actually do something with it, right? So in order for your gene to be expressed, it has to be transcribed, right? You have to make a copy of a piece of DNA into a piece of RNA, which can then become a protein. Or if you're a regulatory element rather than a protein coding gene, you have to be sufficiently accessible to the factors that need to bind you and in the vicinity of the other piece of DNA that you need to bind to regulate it in order to do that, right? So in order to be used, a piece of DNA needs to be accessible to the systems of the cell which use it, right? So to bring that back to the developmental discussion we were talking about earlier, what epigenetic actually does. So, like, which bits of the genome are accessible in use in your brain are somewhat different from the bits that are accessible and in use in your liver, or any of the other organ systems, right? So the developmental process is kind of a process of the body, like figuring out like the order in which it turns on and off all of these things to grow a body and have it specialize into all these different cell types, right? So you have this process wherein stuff is turned on and off epigenetically over time in order to grow and then maintain the final form of the organism, using the appropriate subset to the appropriate degree, and with the appropriate responsiveness to environmental cues of all of the possible tools that are available from the genome. So that's epigenetics, right? Yep. And that's it's another layer of information, right? So that, those methyl marks, they're fairly digital information, and then you've got the histone annotations, they're kind of moderately digital annotation. We've kind of talked before about how biology is like a hybrid digital analog computational system, yes, right? So you've got the digitalness of the DNA, and then increasingly as you get out from that, the sort of control systems of the cell are a bit more like analog computation, right? They're paying attention to gradients and concentrations in a more continuous fashion, though there is still some stuff that's a bit more switch-like. But that information is harder to maintain signal integrity in, as anyone who's worked with analog versus digital signal processing will tell you, right? Analog signal is harder to maintain good quality of than is digital signal, because it doesn't have the same rigorousness of checking mechanisms. Yes. So one of the things that we see with age is not just a degradation of the genetic information, but of the epigenetic state that tells cells which cells they're supposed to be.
And could you, I think this is a great point. This topic, what you just described, Richard, is really, will lead us to more expanded, conversation on things like stem cells and their state, and which is important for the body regeneration. But before we do go to that, could you sort of expand on why epigenome and the reset of epigenome is important for aging? To prevent aging or reverse aging and basically biological immortality.
Yes. So I think the kind of two hallmarks we're covering here are a little bit related in the sense, right? You've got epigenetic alterations and you've got loss of proteostasis, which is proteostasis is like homeostasis, but the maintenance of protein state, right? And because many epigenetic marks are protein based, there's overlap between those two domains, right? And a lot of information resides in the protein space. Just to kind of wrap up that little overview of various sections we've been doing on the primary hallmarks, genetic instability, telomere attrition, epigenetic alterations, and loss of proteostasis. But to return to why the epigenetic state is important, so one of the more recent developments in the aging field has been the advent of epigenetic clocks. So the methylation feature that I mentioned before, this little extra carbon and 3 hydrogens you stick on a cytosine, is relatively easy to measure by comparison with some of the other marks. So the stuff that's protein-based tends to involve a method called ChIP-seq, or its more refined derivatives that are better at excluding background. And they're not as quantitative as the DNA methylation measurement methods that we have, or some of the good ones. So we can use kind of a technology to measure the proportion of cells that are either methylated or unmethylated in a mix of cells from a tissue. And with that, we've been able to construct. So this work sort of originally started with Steve Horvath in 2013, working on just a bunch of publicly available DNA methylation array data, and he fit a big model, an elastic net linear regression for anyone who's interested in the technical details, which just effectively picks a subset of sites that are most predictive of age, right? So what he was doing was taking the methylation arrays from people where we knew the age of the people and we had the methylation profiles, and trying to predict from the methylation profiles what the age of the people were. And the end result was it like 353 or 356? I can't remember exactly what the number was. 350 some odd individual CPGs provided a prediction of your chronological age with an error of about 3.5 years.
Okay.
Right. So, if you give me a methylation array and you don't tell me how old this person is, I can look at those 350 some odd sites, use the coefficients from Horvath's model, and say this person is probably 35, right? And I'm very likely 95% confidence interval to be plus or minus 3 or 3.5-ish years with that estimate. But the more interesting thing about that work was that the difference between the estimated age and the chronological age was highly predictive of lifespan and how long you'd live, sort of all-cause mortality, even when you correct for a whole bunch of other factors, right? So if you put in, smoking and socioeconomic background and like a whole bunch of list of other stuff that you can think of that might possibly impact people's lifespan, and you adjust for all that in your modeling, you still get a pretty robust effect of like the difference between your predicted age and your chronological age is predictive of how long you'll live. So if you're older chronologically than you appear biologically, then you're not in good shape. But if you're younger biologically than you appear chronologically, then that's a good sign for you to live longer.
May I ask, were those 300 CPGs were located some specific areas of the genome, like some certain genes or regions that were of importance?
So the the interpretation of these clocks is a difficult and complicated area. So just to quickly, you used a piece of jargon there, CpGs. So the C's that are methylated are always next to G's, pretty much in biology. There's unimportant reasons for our purposes for that, but the CpG is just like the occurrences in the genome where there's a C followed by a G. That's where they can get methylated. C's in other contexts, not so much. So the particular location of those, like a few of those are in like kind of interesting sounding genes for aging purposes. Maybe they've come up in a few other aging related contexts. But the way that the statistical model which does this works is it tries to pick the group of genes that will collectively predict best, and it will exclude any genes which are highly correlated with those predictive genes, right? So it will try and throw away anything that's not adding additional information to the prediction of the model, which means you may miss stuff which is quite well correlated with age, but it's just not like the best thing for predicting age in this model, right? So when you look at the biological properties of the features which show up in this model, okay, yes, they're interesting, but they're not going to be the only interesting things that might be correlated with age, right? There's a bunch of other stuff that you're not keeping in there. Yeah. And we've also done the equivalent thing of just like fitting a linear model for every single CpG and seeing and looking at what the correlation is, right? All like 850,000 of them that we have on the array, right? We have the models and we look at the slopes, and some of them are better and worse correlates. But there's thousands of overall sites that correlate with age with their change, right? A lot of them have fairly linear changes with age, actually, which is quite interesting, right? So you have a linear pattern of alteration in DNA methylation, but you're seeing a super linear pattern of increase in age risk in other contexts, right? So you have this.
May I ask, has anybody done a experiment where they would look into? I know this is done more in the human lifespan, right? But for example, looking into a simpler organisms and predicting their biological age and stuff like that. But have they also looked into inducing, like, for example, demethylation of those areas and seeing whether there's any life expectancy increase, decrease type of thing?
So it's epigenetic clocks have been made for many organisms now. Mice and rats both have pretty good ones. With respect to actually like experimentally intervening in the methylation levels of like the specific clock sites, that's not really been done because that's very technically difficult still. That requires like individual targeting of the loci.
I was just wondering if anybody thought of like using CRISPR-Cas9 system, whereas like they lead, I don't know, a protein or attach the demethylase protein onto the system and just basically letting it have a ham on those CpG areas. But
I don't think anyone's. I mean, the thing is that would be kind of. Yeah, I don't know if anyone's. No one, to my knowledge, has tried that because that would be a really expensive experiment because you'd need, and a very complicated one because you'd have to do, like 350 simultaneous.
No, I know. That's why
events.
That's why I'm like, but I can imagine the difficulty. But I was just curious if whether they're artificially impeding that cycle.
So what has been done though are like caloric restriction, dietary restriction, and various longevity drug treatments and clocks, right? So you can see that the biological age as measured by these clocks does not increase as rapidly in organisms that are longer lived through some intervention or other, right? So if you take longer lived mutants, or if you give them drugs that we know extend lifespan, or if you like remove senescent cells, I can't remember exactly which combination of those have been done with clocks, but a lot of experiments have now been done where you take a clock for an organism and you do some kind of life extending intervention, and the biological age improves. So it's a good biomarker, as they say, of aging processes. So should we try and continue with our like hallmarks kind of walkthrough?
Sure. No, absolutely. I think this is the perfect place to going from the epigenome to going to more of the stem cell discussion, because this area also part of my PhD's expertise, not so much more because I'm not working on it anymore. But in terms of the fact that epigenome state, plus a lot of other factors, let's be honest here, influence the state of the stem cells in the organism, right? And here, by I mean is that we need to clarify a few definitions. But we've mentioned stem cells before in our previous episodes, and basically these are cells that have certain potency. Now, depending on which stage of cells we're talking about, here let's focus on pluripotency, basically cells capable of differentiating or becoming other cells, but specific to their lineage, right? If we were talking about, like, for example, totipotency, cells that, straight after fertilization of an oocyte by a sperm cell, that's where we're talking about the cells that are capable of becoming any sort of type of cell within our bodies. But these, all in all, we call them stem cells because they can produce basically cells of different type from themselves. Now, the idea of aging and relates to the stem cell population, because with time, as our bodies get older, the population of the stem cells within our bodies are reduced. Usage of the cells, the cycle of the old, we mentioned senescence, and obviously the use of the stem cells. There's a progressing with time, there's a progressively less and less cells that would refill, let's say, replace the cells that have, for example, went senescent or died over time. So this idea of in biological immortality is to reintroduce or reestablish the stemness of certain cells, right? To bring back the state the cells were that would lead to the body's, basically the maintenance of the body for a bit longer, right?
Mm-hmm. And so this is the so-called integrative hallmarks, right? The proximal causes of aging phenotypes. We've got stem cell exhaustion as one of them, and altered intercellular communication as the other. And those two are kind of somewhat related, right? You have the renewal capacity of tissues is somewhat limited by the availability of stem cells to replenish them. So if you run out of stem cells in that niche, then you can't replenish that tissue.
Exactly. And as with time progresses, there's a lot of areas in our bodies, throughout our bodies, but more famous ones are, for example, bone marrow, as an example, where as we age, basically those tissue gets replaced because the replication and copying of the stem cells is seriously impeded. But there is a we could go in a lot of here about the whole necessities behind of, like, what would be required for the redifferentiation of cells, for example, of that already dead into more stem-like, with use of a lot of factors and compounds. But I think we need to define a certain aspect here, which is the Waddington's epigenetic landscape, which is also involved. It also relates to the stem cells, because it's also used to explain stem cells, right? So So I think if
I was thinking exactly that.
So if you could, Richard, give us an example of explanation of what is that landscape, and visualize it for us.
Sure. So it relates a little bit back to the epigenetic hallmarks that we talked about earlier, the loss of epigenetic state, and the origin of epigenetics in a developmental context, right? So Conrad Waddington was the originator of this concept of the epigenetic landscape, where you have the stem cells kind of at the top of this hill, and then you have like a river valley that's coming down this hill. Over this landscape, you've got kind of ups and downs and grooves and peaks. And at the bottom of this hillside, where all of the distributaries from that initial single river in the middle reach is the sort of terminally differentiated state of the cells, right? So you've got stem cells up the top of the hill, and you've got terminally differentiated specialist cells doing their brain thing and their liver thing and their lung thing and their eye thing, all at the end of that landscape. So you have to roll this ball, as it were, roll the balls of the cells down the hill, and they will pick a branch in this tree, right? So the developmental process kind of shapes the balance of probabilities of where it is that you will land up, so that which valleys you will fall down at various points along that landscape, so that you end up with the correct composition of cells at the end of rolling down the hill, right? You have the right number of a given type.
This is this relates to both the epigenome and the state of the cells, because obviously these are basically very linked together.
Yeah, and you can kind of the stemness of the cells can be traced up that hillside, right? So you have the totipotent cell at the top, you have the terminally differentiated cell at the bottom, and then at higher branches in the kind of tree of that river, where you have several distributaries branching out, you have the multipotent and the pluripotent stem cells as you go further up that landscape, right? So the potency, as it were, is also represented in that space. And so it's a useful sort of mental picture to assemble to reason about this problem. So some cells are kind of like one of the niches, as it were, is actually not to roll all the way to the bottom of the hill, right? It's to retain a few cells in the stem states a little bit uphill, as it were.
I would say like little lakes on the way down to the very bottom of the ocean, basically to maintain those, because these are crucial for us to basically replenish anything that's, for example, given the damage to the tissue, whether mechanical or otherwise, and even on general, like your skin, for example, it needs to be replenished every day, because with time the keratinocytes and epithelial cells on the surface will die, but they form a dead layer, which basically allows you need to replenish it over time, right? And there's other concepts related to this with these cells. For example, like with aging, obviously you can tell, speaking still of skin, things like less extracellular matrix being deposited by some of the cells, so that your skin becomes less and less elastic, ending with a lot of wrinkles, type of thing. But the reason why it's so important to talk about these cells in terms of biological immortality is that, as with a lot of things before we mentioned and discussed, that there has to be a restart, especially of those cells, without pushing them too far, right? So we need that reactivation of the stem cells with the aging population if we want to prolong the life expectancy of the population, but without leading ending up with a errors of like pushing the cells too far, for example, to their almost near totipotent state, where you could end up with things called teratomas, where basically you have random cell populations, type of cancer where you have random cells populations within those because of the stem cells going a bit haywire, right?
One of the sources of certain diseases, like particularly certain types of blood cancer, are stem cell niches that kind of go a little bit off track, right? Yes. They start producing clonal cells that are disproportionate to the amount that they might need, and there's kind of this like subcancerous versions of this where and this may not be a blood cell type specific thing. It's just one where we notice it most so far, is that the stem cells produce kind of a too many of a clonal copy of a particular stem cell, right? And that can be a problem in itself, but it can also to some degree contribute to the loss of that stem cell reservoir, the stem cell exhaustion, right? Because this stem cell's gone haywire rather than actually doing its quote unquote intended function of providing some replacement functional cells.
And here, we face this issue where, in a way, a lot of research that's done on stem cells and stem cell regeneration is that the use of, for example, certain compounds that or factors that would lead to reversal some of the states of the cells, but which is ultimately, basically, in a lot of cases, reactivating certain genes that are silenced by the very by the very thing that we just discussed, the epigenome. So the fact that some of the factors that were originally expressed in our cells.
So I think we should probably just step back slightly and reframe this in the context of induced pluripotent stem cells and rejuvenation by inducing stemness, right? Do you want to talk a little bit about the iPS cells?
Sure. So we've mentioned this before, and we talked about the idea of replenishing the stem cell population, but quite a lot of research nowadays looks into a formation of stem cells from already differentiated cells. The most famous example of the research is obviously the fact of use of the use of Yamanaka factors, which forgive me, but I can't remember at this very point. But I'm sure if you watch our previous episodes, the references, I'm sure they're somewhere there.
I think it's an Oct4 notch something or other, CMYC,
and SOC something.
Basically,
there were several factors identified that if they're introduced into the cell, the cell populations start to behave and express the sort of morphology of the cells becomes stem cell-like. And this pioneering research showed that those cells that we are actually capable of reintroducing the stemness to a already differentiated cell by providing certain factors, like Yamanaka did. Now, of course, with time, as the time research progressed, obviously we've noticed there's depending on the cell population, depending on there are a lot of factors. Things can induce the stemness, and not just factors like protein factors. Some short RNAs like microRNAs are capable of, few nucleotide long RNAs are capable of that. Obviously, as we mentioned, the epigenetic state of the cells has influence on the stemness of the cells. So it's a way more complex system than obviously we are describing here, but with time, we are understanding more and more of this idea. Now, having the introduction of the factors now is limited to the fact of the localized delivery, right? If you can imagine, if it was a pill given to you, don't think it would be a great idea, because the delivery factor to your specific areas would not be that efficient, and that's why
you wouldn't want to de-differentiate all of your cells and turn them back into stem cells, right? That would be you turn into a pool of mush.
Exactly. So you would that would not be the ideal place. So here, this is why this is the biggest, I think, one of the bigger limitations of the whole reaching biological immortality is that the fact that if we need to find the ways to be able to introduce this pluripotent or stem-like state in the cells by localized delivery of certain factors, as I mentioned earlier. So it's a quite, I would say, big issue. Not obviously not saying that other things are not an issue, because a lot of things, this is just one part of the bigger complex system that is involved in the aging process. But it's one of the ones that relates to a lot of other things that we mentioned today.
So there's this concept of rejuvenation through like rolling your cells back a little bit, right? And getting them a little bit more stem-like, and then having them redifferentiate into new functional adult cells. Right? That's one of the kind of the prospects in rejuvenation-like therapies, and it has had some moderate success in some test organisms in mice and rats, where they've done like a partial de-differentiation of cells and then allow them to redifferentiate by kind of briefly turning on these Yamanaka factors in I can't remember if it was organism wide or in a subset of cells, but it may have been like a tissue specific induction of those factors. I forget. I'd have to go look up the research, but there's some initially promising results, but I think they also had some mishaps of now we've given them the rats and mice cancer variety, so it's a challenging thing to do, and this so one of the other kind of just to talk a little bit about some of the current discourse on the subject. There's a few popularizers now of the scientific study of aging, and David Sinclair is one of them, and he talks quite a lot about this concept of like rejuvenating tissues through the like de-differentiation of cells and having them roll back down again. He also has this epigenetic information theory of aging, right? So he shares my perspective on that, right? The importance of epigenetic information in aging, but where we have a kind of a slight difference of opinion on this point is that he seems to think a little optimistically, in my opinion, that if you just roll the stem cells back up the landscape a little bit, right? If you just let them de-differentiate a slight bit and then let them roll back down again, that they will end up falling into the correct grooves, right? Firstly, it's like, can you even successfully get them back up the hill in the appropriate way?
Exactly.
Right? Does actually, we don't really know if like, does turning on the Yamanaka factors like actually produce a cell which is kind of just a multipotent like muscle cell that can just do muscle stuff, or does it give it some other inclination to potentially end up going down the wrong differentiation path, right? So you need the fine granular control over how the cell de-differentiates and then how it redifferentiates again. And the question that kind of that my difference with David Sinclair might hinge on this point is the epigenetic information like still present in the cell if you de-differentiate it a little bit, or is it available to the cell to redifferentiate correctly from the environment, right? We still don't kind of really have a firm grasp of how much of that information is still available and how much of it can be shifted by the environment, right? It may be, and it would be nice to know that it'd be nice if it was, that a lot of that information is still present and we would get back a cell that's very much like the desired adult cell type, right? And I suspect that to some degree it's there, but I think the degree to which that information is kind of accurately represented is going to be a problem for the effectiveness of this approach.
Absolutely, I absolutely agree with you in here. I think it's quite an optimistic approach because simply in a lot of research, depending on what specialist you talk about in stem cell regeneration, is that there's still disagreement in a lot of aspects of cell de-differentiation and then differentiation of stem cells specific, right? So if we want to have cells, for example, the easier part, right, is to roll them down the hill, right? We can provide them with a cocktail of things, but we are still uncertain on the aspect whether we are actually doing the correct job, right? And if I may, I could give you an example. This is more of a personal example where my own research involved bone regeneration, right? And we would use stem cells, bone marrow stromal cells, so cells from bone marrow that exhibit multipotency or pluripotency, depending on how you look at it. And you can guide them to either become a bone tissue, a cartilage, or for example, an adipocyte, so a fat tissue, right? Because they are related, right? And we still and there are still arguments on the fact of using certain compounds and factors to, for example, create a bone, right? And in a lot of aspects of the stem cell, like stem cell regeneration, requires a more compound thinking in terms of, as Richard mentioned, does the epigenome of the cell change? Or does it not change at all? Or is it something affected? Is it affected somehow? But in what sort of state? What changes are there when we introduce, for example, differentiation or de-differentiation compounds, depending on which way you want to do it?
I mean, if we return to kind of the original developmental context in which these differentiation processes happen, right? The cells are taking cues from their environment.
That's what I was going to say as well.
What to do. Yeah. So the things like gradients of different chemicals in the structure of the body, right, will tell. Did you want to go into that?
No, this is exactly. I was going to exactly mention that the fact that but the limit is as Richard just mentioned, is the environment, and we are talking about not just a 1-dimensional, 2-dimensional. We're talking about 3-dimensional, and actually, in fact, 4-dimensional because of including the time, including of the gradients of the compounds and cytokines and all other things that are surrounding certain cells within the tissue and and organs. So. A lot of that aspects are still not fully understood, simply because we look into, obviously in science, we break things apart. So we take one step at a time, like one variable at a time. But realistically, all of those things interact with each other in a very complex manner. So the fact that, trying to push a cell up the hill and then, believing that it will go the way, where it has to be, is quite a, I would say, very, almost, I would say, too optimistic way of thinking. Simply because we don't know what sort of environment we've created by, for example, forcefully inducing a pluripotency or multipotency, because the cell, for example, may get different gradients of compounds or cytokines surrounding from their environment, and then go complete different direction that we expected.
To just sort of to contextualize that, so when a cell is originally developing, there's a lot of patterning done in the developing embryo to kind of effectively tell cells where they are in the body plan, right? So you have gradients of chemicals, like morphogens is sometimes what they're described. There's a bunch of different stuff like that. Where let's say, and as a sort of crude oversimplification, let's say I've got a head to tail representation of the body, and I have a chemical that's a high concentration at the head and a low concentration at the tail, right? And then if I'm a cell kind of 3/4 of the way back in this body plan, then I'm getting that concentration is associated with starting to do stuff that might be involved in developing legs, right? Whereas if I'm roughly 1/4 of the way down from the head, I'm starting to do the arm related development, and all the way at the head end I'm doing head stuff, all the way at the tail end I'm doing tail stuff if I have a tail.
Absolutely.
Depending or not. Yeah. And there's kind of intersecting gradients of these chemicals, right? So you might have a counter chemical where you've got the opposite gradient that's concentrated at the tail and low concentration at the head, right? So the intersection of those two things provides information that the signaling can be useful for, right? So the information that I'm roughly 75% back in the body plan, so I should do legs, is kind of a bit fuzzy, right? There's some room for error there. But if I add into that the additional information that I'm 25% from the back from the other chemical gradient, right? I have the cross section of those two gradients. Then I have more confidence that I should be doing this thing and not this other thing, right? I have additional points of information that I can use to integrate and figure out what I should be doing, right? And developmental processes are full of information like that. There's all of these complex intersecting chemical signatures that tell cells specifically what they should be differentiating into as a part of the developmental process. And what's a little bit unclear at this point is to what degree the information which is encoded in those chemical gradients that may be ephemeral during development is still present in the adult organism, right? To what degree can a cell that is in my hand infer from its environment that it should be in my hand, right? Is the information there necessary to tell it to still be a hand cell? And there is some sort of promising stuff from that, right? From organisms which are capable of regrowing limbs and the like, right? So that capacity is clearly there in those organisms, right? So if you take something like an axolotl, which is a particular amphibian thing, and you cut off one of its legs, it'll grow a new leg pretty effectively, right? So like whatever the cells were that were, you know, around the joint where you cut the leg off, know that that's where they were, and that the cells that were adjacent to them have gone missing, and what they should do to replace it, right? They can recapitulate that developmental program that happened when the organism was originally developing to generate a new limb. And there's like some degree of evidence that can be attained partially, at least, in humans, right? With the right particular cocktail of chemicals, you can start to get like digit regrowth in mice and stuff like that, right? So there's some latent potential for that representation of, you know, where I am in the body plan and what my cells should be doing to still be there. But it seems like it's not as precise in the adult organisms and not as accessible to the cells, at least in the mammalian body plan.
It might be. I feel like this might be related to the fact that that there's this gradient still present in those amphibians, right? For example, in their being expressed, so the cells continuously get. But also, I think it's related to the size of the organisms, often that we talk about that have these capabilities. Because I have this feeling that we could induce, for example, in amputees, for example, someone losing their hands or legs, that we could induce the cell regeneration of the limb. But I feel like it most likely would be of the state of when we are newborn children type of thing, where basically we can get this, but the progress where we get to the complexity of our body where it is due to the time that spent, the growth and etc. So I feel like this is an issue where, one, the lack of the gradient, and two is that we lack of the 3D cues or in the 3D environment that was originally there, right? Mm-hmm. And often I think is also because of the sheer size of what we try to achieve. And I'm not just talking about like the grandiose size. I mean physically mean the size of our bodies, right? It's so much easier to do something in a much smaller body, which requires less complexity.
And I think a component of it may well be evolutionary, in that there are trade-offs, right? So if you look at the environment in which some of these organisms that can regrow limbs exists, like they will frequently encounter situations where they might lose a limb, right? So it's beneficial for them to be able to grow them back, right? Whereas for us, the trade-off might be different, right? Maybe the capacity to do wound healing of a more localized nature is in some way in tension with the ability to completely regrow a limb, right? Is it like, can you retain like the structural integrity of a wound and form like an okay job of healing it when you're in an environment that's kind of more stressful, right? Do you need an environment that's kind of able to facilitate the regrowth of that limb? So a lot of these animals are aquatic, right? The ones that can regrow limbs, right? So that they're not on land in an environment where like the limb is subject to more stresses, right? Just gravity, effective gravity is different for an organism that's on land. Whereas in water, right, if it's kind of neutrally buoyant, the new limb can just kind of float there, right? It's not in so much risk of being a problem for that organism for the kind of fragility of that limb in the intermediate regrowth process, but for a a land mammal, right? It's a problem if you're, kind of regrown a baby arm and it's a bit floppy,
Yes.
right?
I mean, that explains, like the whole survivability, and also, it's this flight or fight, system. Like, in aquatic environments, you said, like, if you lose a limb, you still have 3 other ones that will help you to escape. Whereas in humans, you lose a leg. I would like to see you jumping away from danger. So I think this is the big problem of, like, one of the problems that would have to be tackled for in terms of, if we want to address biological immortality, is the fact that the understanding of the stem cell state, being able to push them back, the understanding to that up the hill state, understanding what changes are in the epigenome and what effects does they have on the epigenomic state, and then whether we can successfully lead them to the appropriate valley with a localized stimulation with factors without causing without stimulating, for example, formation of a foot of a toe in your brain, for example, because your factors have stimulated your neuron cells to go in the wrong way that they should have.
Well, it's kind of like there's 2 approaches we could take to that problem, right? There's the kind of like the cultural one of, like, develop the medical technology necessary to figure all that stuff out and try to recapitulate it, and then we could also do the kind of the genetic engineering route, right? We could try and figure out how to make a revision of our genetics that gives us these abilities natively, and address the trade-offs that evolution made, that we may not want to make, because the environment in which we live now is no longer quite like our environment of evolutionary adaptiveness.
So, Richard, is there any other hallmarks you would like to talk, or should we talk all other sort of binary things that we sort of thought of that are involved in, would be involved in biological immortality, important for biological immortality?
So let's quickly wrap up the hallmarks thing. So we just did the integrative hallmarks, and we started out with the primary hallmarks. So the remaining ones are the antagonistic hallmarks, which is dysregulated nutrient sensing, mitochondrial dysfunction, and cellular senescence, right? So we kind of covered cellular senescence. I don't know if we mentioned the effectiveness of removing senescent cells from adult organisms, which has been shown quite nicely in mice, and is a big area of research focus in drug development in the kind of longevity and
we never really mentioned that, but it's currently quite a big topic to, like, removal of senescent cells or reversing their senescent state state. But at the moment, as it is, it's still a area of research that is quite prolific, I would say. Quite a lot of people are starting to work on these things, right? But I think the problem is that the question, I personally think, is that the question is with the problem with the reversal of senescence of the cells is that if you do reverse the senescence and the cells start to replicate, what was the original cause of senescence, right? So...
Well, I think that's the goal of most of those treatments is lysis, right? They want to break up the senolytic cells rather than return them to.
Well,
to
you
a
say that,
state.
but, like, for example, back when we were doing PhD, like, quite a lot of there was a research done by one of our coworker, Becky, if you remember her. She did on reversal of senescence from gingival tissue.
Okay.
So that was and that's quite a big topic as well. Not like be able to actually reverse senescence, right? So
interesting.
Yeah.
I didn't know that much about the reversal of senescence.
Yeah, it was actually quite interesting topic because the way they looked at it, she looked at it, was basically introducing a compound that was found to be able to actually reverse senescence, and in fact that as you mentioned earlier, the senescence markers or the cytokines that the cells were releasing were actually seized by the cells, and they actually started behaving more of like a stem cell like behavior.
Okay.
Yeah, but you mentioned other hallmarks of the aging. Earlier on, just now. So, the nutrient.
Yeah, so then we got the dysregulated nutrient sensing, right? So a lot of that is insulin-like growth factor signaling related stuff. All the things that are integrated through the target of rapamycin signaling complex. And sirtuins and AMPK and all that kind of signaling about whether there is an abundance of resources and we should be doing lots of anabolic and biosynthetic processes, or whether or not we should be if there's an absence of resources and we should be in responding to a state of scarcity and adversity. And there's a lot of aging-related interventions. In fact, so the first life-extending phenomenon that was identified kind of scientifically and systematically was this concept of dietary or caloric restriction, where you basically feed an animal or anything from a yeast cell to a C. elegans worm to a rhesus macaque, right? You feed them less calories and they live a little bit longer, right? The effect is often larger in some of the smaller and already less long-lived animals, but and that relates to kind of a concept that we've not necessarily covered as much, which is hormesis, right? So the notion of hormesis is that you have a response to stress that is like increasing your robustness, right? So it's a bit relating to the kind of antifragility concept, but it's not quite antifragile. It's still a bit more in the robust space, right? So you if there's some stressor, then you might dial up your resistance phenotype, right? So I'm in a resource scarce environment. I'm not necessarily going to be able to reproduce immediately. I need to knuckle down and take care of what I've got, right? That's the kind of general attitude that I'm getting from this whole response, right? So you take by inducing one of these stresses through a slight lack of nutrients, right? But by making the organisms think they're in a state of stress, in a state of scarcity, they dial up the pathways that they use to stick around longer and stave off their own decline until they can reproduce. Right? That's what we think is the kind of general underlying mechanism of this dietary restriction type phenotype, which is incredibly widespread across all organisms.
Interesting, if I may say, because a lot of recent sort of studies shown that like a regular fasting or fasting every so often actually does help for the maintenance of the body, just like for the prolonging your life, as they say it. But like basically the idea is actually gives the time for the organism to sort of regenerate.
That's the idea, right? It's it's sort of dietary restriction mimicking or diets or compounds, right? Compounds which trick the body, or behavior patterns which trick the body into thinking that it's in a situation of nutrient scarcity, and thereby dialing up these pathways that are pro longevity in nature, right? So you might turn up your DNA repair and various other your proteostasis. You might dial that up and dial down some of the stuff that might cause damages. A whole host of different things sort of shifts, right? You have there's this kind of two modes, right? There's the everything's good, let's go. Use the resources, reproduce now, and then there's the things are not so great right now. Let's hunker down and wait for a better opportunity.
Absolutely. But speaking of diet, I think this is just briefly. We should probably mention this, that since we are on the topic, is the fact that diet itself has a substantial, obvious effect because on our life expectancy, right? The standard reduced salt, sugar, fat, right? Like the whole salt, because of it leads to arterial closure and plaques that will lead to heart issues. Sugar, obvious, like the whole idea of sugar, more sugar than fat. I'm sorry, I'll clarify, because sugary foods lead to things like fat cells to expand and basically change their morphology and expression, cytokine expression, which leads to a lot of other complex. We meant, this is quite,
and even just like glycation of proteins from high blood sugar
so
levels.
There's a lot of problems associated with certain diets, especially nowadays with easy access fast food, stuff like that. Red meat consumption type of thing, where like a lot of the countries, for example, like in America, where there's a high level of red meat consumption, leads to to cancer because of indigested proteins and stuff like that. And we mentioned things like compounds that help us, like for example curcumin, right? We've talked about it, and we showed like that's basically research been done, and basically it's like a miracle thing that basically you should have a spoon of it every day, because it just affects everything.
More popularly known as turmeric, for anyone.
And we mentioned this in episode 3, just of Xenothesis, when I've checked down, because I couldn't find resource. I was like, the reference to the turmeric, I was like, and then it literally is episode 3 when we talked about it. So those things we've mentioned and talked about, these are very general aspects compared to what we just talked about now, but these have a substantial effect as well on the longevity of the life expectancy, right? Not only that having a access to food, to nutrients, is important in general. Obviously that as Richard mentioned earlier, reduction of intake of protein, of nutrients in general, is good to sort of prolonging, but an excessive lack of nutrient access is detrimental, obviously. But obviously this topic will require another, I think, 2-hour recording explaining all of this aspects. So in here we just briefly mention them.
Dietary science would not even, there would be it's still a very unsettled in many ways aspects of that, but the one universal that came out of all that was if you eat less than your calorie intake, then your recommended calorie intake, right? Your minimum, not minimum, but your if you eat just below that kind of level, then it actually has a fairly substantial measurable impact on your lifespan. And that was kind of as a point in the history of biogerontology, right? The history of aging research as a discipline it's a fairly important milestone, because it's actually a scientific result that's very widespread in different organisms, and is highly established, that there is an intervention that you can do that will actually impact how long organisms live. And the kind of next major milestones in that space were the identification of single gene mutations which have an impact on lifespan, right? So we've been talking about the complexity of this system, but there are small changes which substantially impact lifespan, but all of which have some kind of trade-off, right? So in C. elegans, you have the famous DAF16 mutant and the AGE1 mutant that extend the lifespan of the worms by a very substantial amount. But if you then put those worms back in a population of ordinary worms in the environment, as it were, in something that's a bit more like where they would normally live in the lab, and even just on a lab dish, right, they will be outcompeted by the normal N2 strain, right, the wild type strain that we use, because mostly because they're not as reproductively fit as the wild type, but there's some trade-off in that gives them additional longevity, but it costs them a little bit somewhere else.
And we mentioned that basically.
Although that's not necessarily it's not necessarily the case in all cases that there will be those trade-offs.
Yeah.
There are some things where we can, and of course there may well be trade-offs that we're willing to make to some. It's just trade-offs that evolution wasn't looking to make. It's the point there.
But yeah, so going back maybe to the finishing of the hallmarks. So we had the nutrients, we had senescence, we had the epigenome landscape. There was one more thing I think you mentioned that I'm sorry, but I can't remember.
Mitochondrial dysfunction is the last one on the list that we've not really covered yet. So for a while there, the reactive oxygen species theory of aging was pretty popular, right? We accumulate these highly reactive compounds that just go around doing damage by breaking stuff. And the mitochondria are the primary place in the cell where these reactive oxygen species can be produced. So if you have a slightly dysfunctional mitochondria, then it may be producing an excess of these reactive oxygen species from they're a byproduct of respiration, and they're in the electron transport chain. You get these free radical compounds that need an extra electron pair. So the thing with that whole reactive oxygen species theory was that it didn't really bear out, right? If you take all of the if you really dial up the amount of reactive oxygen species in the worms, for example, right? If you delete all their SOD genes, right? SOD is this superoxide dismutase, is the enzyme that cleans up all of the reactive oxygen species. Then they have a normal lifespan, right? Doesn't make them live shorter. So they accumulate a lot of additional oxidative damage, but it doesn't necessarily actually kill them off
straight away.
Much more than other stuff, right? Yeah. So it's not really impacting the lifespan that much, and the opposite, just dialing it up doesn't really have that much of an impact anyway. And part of the reason for this is reactive oxygen species are a thing that we've had to deal with in the environment, right? The cell has homeostatic mechanisms for handling them, and it has defense in depth, right? It's not just the SOD genes. There are other things in place to deal with reactive oxygen species damage. Indeed, immune cells will even use them to some degree as offensive weapons against bacteria and stuff, right?
They're used within the cells to break down certain compounds or certain things. So
yeah. And so actually, one of the things I didn't mention when we were first covering the hallmarks is the hallmarks all have to meet the criteria of manifesting during the normal aging process, accelerating age when they are aggravated, and retarding the aging process when they are ameliorated. And so it doesn't. In order to qualify as a hallmark, you have to meet those criteria, right? You have to show up when you're normally aging, make aging worse when you're dialed up, and make aging better when you're dialed down. So the ROS thing only kind of doesn't entirely meet that, that criteria very well. Although mitochondrial dysfunction is still, in some other aspects of it, an interesting problem, right? That there are limits to the degree that we can cope with excess reactive oxygen species, and there are problems with other aspects of mitochondrial maintenance, right? Are they providing sufficient energy for the cell to do things? How's their life cycle working, right? Are they being disposed of through mitophagy effectively and being replaced well? And do they have are there weird mitochondrial mutants with suboptimal phenotypes accumulating because they're no longer subject to kind of the sort of the selection process that they would have early on in the germline, right? So there's a whole bunch of stuff that could be associated with mitochondria that's still potentially a contributing factor.
I see. So I guess this basically summarizes the hallmarks of aging, right?
Yeah, that's all of the hallmarks.
Awesome. I mean, it's 2 hours now where we've been recording, and we've managed to, but there's still a few things that are actually can affect, I think, the biological immortality, that the few points that we sort of came up with, that still sort of, I think are worth mentioning. We talked about stem cells, but there is some certain cell populations within our bodies that are important for us, but they don't really, after a certain age, they don't replicate anymore. And I'm here specifically talking about neurons, and the maintenance of neurons and prevention of their degradation is one of the I think, problems that is also associated with the stemness, where basically to reach biological immortality, we would have to have the ability to sort of maintain the neurological state that we are at our peak without the loss that's associated with aging, right? And with time, as you know, things like debris accumulation in our brain, the lack of regeneration of neurons, and the reduction of maintenance of the neurons, it's what basically leads to degenerative diseases that often are associated with aging, for example, dementia.
And the kind of conventional approach to like healing of a tissue is a bit of a more challenging undertaking in the brain, right? Because the structure of the brain, what connections an individual neuron has to all of the other things, it encodes the information that makes up our consciousness.
Exactly.
Right? So we can't just go in there and just like, okay, we're going to replace all these neurons without actually, carefully replacing all of their individual connections first, because otherwise we just, okay, maybe we're immortal, but we've completely reset our brain structure every
becoming
now and
completely different character type.
That's not that's not really continuity of consciousness.
Exactly. And this is the thing, like the idea, we would have to have a system where as we are growing as children, right, our brains still are producing more and more neurons, right? And at some point that process stops. So we would have to have a system where basically allows for a reconstitution or replacement of the cells that we've lost, without obviously the leading to the effect that we've mentioned in stem cells that could be detrimental to going too far with it, basically.
Yeah, but it's just kind of a similar problem, but even more difficult.
Yes,
right?
I think from all of the tissue, I think obviously brain is the most difficult to work with, simply because in the bone, for example, it doesn't matter what sort of cells, how many cells you have there, as long as they do produce that extracellular matrix that will become a bone. Here we have specific connections, cell connections, that have to be there because they defined who we are. And that's, in a way, it's a pretty difficult, there are cases, when people had, brain surgeries done and they remove chunk of the brains and nothing really affecting their life, no losing any memory or any of like motor functions or anything. There are some cases when people, got hit in the head and completely had a personality change. So it's a concept that it's probably one of the most difficult tissue that we will have to work to address the whole idea of biological immortality, to be able to address those issues.
Yeah. It's I think the most profoundly challenging one, and the one that matters most to us as individuals in that sense, right? Yeah, but I think a component of being immortal in the kind of in the sense of actually living more or less indefinitely requires a certain embrace of change, right? You have to be able to, even as we are now, right? We are different people in some important sense from who we were in previous points in time, right? We grow and we change over time, and that process would, if we're doing immortality right, continue indefinitely, right? We should retain the plasticity and the ability to change. Maybe we try and keep as much memory as we can going back for a more extended period and learn more from those experiences, but we would change as people, right? To be an immortality where one is static and not capable of growth is not one that is terribly interesting, right? It's the subject reminds me a little bit of some of the themes in one of Octavia's other books, actually, in her parables books. In the parables books, I won't spoil anything, but there exists a religion called Earthseed. It's kind of a religion. It's also in a sense it's somewhat secular, but in the Earthseed religion, God is change. And the only lasting truth is change, right? That's the one of the refrains of the religion, right? They're interested in, they have a sort of almost stoic attitude to the fact that change will happen, right? But they're also interested in shaping the direction of that change, right? How can we shape the change to be in a way that we are interested in the world being, right? How can we direct the change? Which is inevitable. That is, I think, also part of what it would mean to be an immortal consciousness, right? You'd have to embrace the possibility and well, not even really the possibility, but the necessity of ongoing change.
But so before we actually, this is a very important topic you just mentioned, Richard, but I think before we go into that, because this will sort of lead us finally to the finish off with the biology behind and lead us more to the socio-economic sort of effects still related to biology. I just want to mention one last thing that could be related biologically wise to the biological immortality, if I may, before we continue with that concept, because I think this will really lead us, what you just mentioned, to the further conversation about the rest of our episode. So this was given by my wonderful fiance, the idea of age-related loss of cohesion. That could be related to the problem with the biological immortality. And let me sort of explain here, what do I mean by that? Aneuploidy is one of the leading genetic causes of birth defects due to errors in the chromosome numbers from oocytes. And this aneuploidy in oocytes increases with advanced maternal age. And basically, recently studies have started to support the hypothesis that cohesion deterioration of those, when formation of the chromosomes is with advanced maternal age represents a leading cause of of the age-related aneuploidy. And the cohesion process is established only during the premeiotic S phase of the fetal development. And basically, it's not replenished anymore throughout the female periods of fertility. And what it does is basically it holds the sister chromatids together until the meiosis resumes at puberty, and then the chromosome segregation requires the release of the sister chromatid cohesion from chromosome arms, and basically, undergo the rest of the cycle.
Okay, so just to clarify, so this is the association of the chromosomes in oocytes. In the oocytes, right? Yeah, so it's the Sorry.
Basically what it basically means is that in women during development of their oocytes, there is this attachment, cohesion of their chromosomes, right? And at some point with that deterioration of that cohesion, that connection, is leading to chromosomal defects, basically. And this is related and this happens more and more with age, right? That's why you don't get like in men, sperm cells get replenished all the time from stem cells. In women, what you get from a number of oocytes, that's what that's what you all get. Like that's the number you'll have to the end, right?
Yeah, and this is why the females contribute more like large structural mutations or aneuploidies. Yes. We have the wrong number of chromosomes, and males contribute more point mutations, right? Small errors in copying in the DNA as we age, because the egg cells are non-replicative, right? You get a fixed number and they stay maintained throughout the life of the woman, whereas in the male, the cells are produced a lot. There's more scope there for introducing small errors in copying.
So going back to what Richard mentioned, the whole balance of between getting reproduction versus longevity, and this is the issue where we are facing here now, is that if we were to prolong our lifespan, it would mean that this process would have to be reversed, and also reversed to ensure that, if we don't have offsprings in the early stage of our lives as we do now, it would have to be, this issue would have to be resolved, right? Because otherwise we would end up with the population where you can live longer, but the population is not replenishing over time.
Indeed, yeah, we would have to extend the window of female fertility substantially.
Yes, basically.
Ideally.
And I mean,
there's already quite a lot of attention
on that. Yes, exactly. And I mean, to be fair, like, once in the age of, puberty, when these women start having their menstruation cycle, now it goes until age of what? Nowadays, 40, 50, right?
I think menopause is normally... I can't remember what the distribution for age of onset is, but I think it's generally 50s, something like that. But then, although the before you get to menopause, there's a dip in reproductive peak, kind of after your, I think, like early to mid 30s, if I remember correctly, something in that general demographic range.
No, but the reason why I'm saying is that, like, now imagine when we prolong our lifespans to 200, 300 years initially, and that menstruation cycle being prolonged for that many decades, relative to, so I just feel sorry for women.
Yeah, I mean, we're now getting into an area of quite different, kind of more like social factors, right? And it's, I think it's a good transition for us to try and make around this point in time.
So I think at this point, we covered most of the ideas we thought that would be, related to the biology, the hurdles in biology that would have to address to reach the biological immortality. But now we wanted to sort of discuss the socio-economic issues related, the pros and cons, if you could say, related to biological immortality. And I just wanted to start with the conversation that Richard was talking about just earlier on, before I mentioned the age-related cohesion, about the whole idea of the change, right? So in fact, evolution in itself, there are certain aspects of it that affect the social and economic, especially economic sort of aspect, and also the energy expenditure, as Richard mentioned, like the use of energy and requirement of energy within the population and an individual to maintain an organism, right? And the idea is, in the social economic status, immortality would lead to expenditure of a greater necessity of resources expenditure on an individual, right, or a population, biologically immortal population, right? Now, in my view at that time, before having conversation with Richard, I thought that eventually the use of resources, the great increase on the use of resources and necessity of use of resources would lead to loss, basically having used the whole of the resources available on our planet to basically maintain the population. But then again, now with the conversation talking to Richard about the whole idea is that if we prolong our mortality, right, the idea would be probably that the use of energy, the lack of necessity to reproduce, would probably balance it somehow. Although I'm not certain it would be.
Yes, I think I see what you're getting at here, and I talked before about, if you want to maintain the integrity of a signal perfectly, you need like infinite energy expenditure, right? But that's not exactly what we were trying to do because of the change, right? We don't want to maintain an exact copy of the signal indefinitely. We just want to maintain a state of the system which is capable of like monitoring and reflecting and maintaining a version of itself which it recognizes as self, right? It's changing over time, but it is not attempting to indefinitely maintain a perfect representation of a single state. So I don't think that it's quite the same energy constraint problem. I think it's a different type of problem, which may may require. Well, as I say, it won't require infinite energy to maintain with exact probability the exact same state, but it will still require energy to move forward. Yeah, but as a general rule, though, like I'm not particularly enamored of the notion that we should try and aim for like a steady state of energy usage anytime soon, right? That we have an entire light cone of the universe to get to, and we're still on one planet, right? If you look at the kind of the concepts of like levels of civilization, right? Using all of the energy available on your planet from your sun, from your galaxy, right? We haven't got even remotely close to using all of our available energy, and there is much more that we could extract. And quite besides that point, right, the history of human development is told in the story of the use of additional sources of energy, right? The gains that we get from civilizational development, right, it is using more of the energy that's available to us and in more efficient ways. That's like use of more energy, as far as I am concerned, at least for the foreseeable future, is a good thing, right? Even in the context of the current climate situation, right? We should not be aspiring to reduce the amount of energy we use. We should be aspiring to increase the amount of energy that we're using, but through means that are sustainable in the long term, right? We want people to have access to energy because that means quality of life, right? More energy to expend, energy abundance is good quality of life. So the means through which we solve the climate crisis, that's a whole other debate discussion, but which we can shelve. But like, we should not be attempting to limit the amount of energy that we're using going forward, except in the extreme long term when we run out of available nearby universe to expand into.
The reason why also one of my arguments is also completely out of the window is because now having talked to Richard about this is like I basically put down the whole idea. I put it because of the use of resources, using all possible resources leading to widespread death because immortality can't be maintained anymore, or changes in the environment, introduction of new bacteria, viruses, pathogens, change to more toxic environment would lead to humans not being able to adapt, right? But this is not exactly correct because from what we've discussed at the very beginning, Richard was talking about the robustness versus antifragility. So the whole idea of this argument is out of the window because the whole idea is we don't want change not to happen. We just want it to be able to happen fast enough to be responding to changes in our environments, right? And that's what would really lead to biological immortality. But yeah, I thought this was one of those arguments that now we've talked. I was like, yeah, this argument is completely out of the window because. Yeah, you very explained the whole concept and where we should be aiming at. But there are sort of, in a way, other aspects that or different types of immortality, if one can say, right? Of course, we decided to talk about biological immortality, but here we can also, in our previous special episode, as we mentioned, in the cyberpunk, there's other things like technological influence or that would prolong our lifespan, right? Like in case of for example, any of our organs failing, we could, for example, in future exchange it with a technological equivalent, for example. And the whole idea of our bodies, if our biological development is not fast enough, technology could overtake it temporarily as a means to an end. Right,
and so I think an interesting point of transition in that kind of transhumanist picture that we didn't quite really get to. But one of the things that, like, a lot of the pieces of fiction that exist in the space of immortality involve like mind uploading type technologies, right? And that kind of is a bit of a response to the profound perceived biological challenge of having your brain live on indefinitely, right? Which we kind of ended up with at the end of our biological aging discussion was like, the brain's really hard.
Exactly.
Right? So one of the responses to that is, okay, well, maybe we'll freeze people for a while in the meantime, and that's the whole cryonics thing, right?
Yeah.
But then we'll figure out how to, scan their brains and upload a version of them to some simulated environment and run them as an emulated consciousness. And that's one of the potential routes to immortality, which is an extremely technically challenging undertaking from several perspectives. Like the instrumentation necessary to get a sufficiently low-level representation of the thing that you're scanning in the first place, the capacity to run a physics simulation at sufficient granularity to actually emulate that scan, even if you have one in that kind of detail, and the ability to capture the kind of dynamic state of the brain, because right, a snapshot of the current network state, right? If you want to take a higher level abstraction of the brain and not like individual protein or sort of subcellular level representations, if you want to try and abstract it to make the computational challenges easier by just doing like a network representation, I don't know if that's going to be enough to actually emulate the consciousness well. Plus, it doesn't have baked into it the capacity for how the network structure should change, right? What are the mechanisms by which you have the dynamics of the network structure change? Because in brains and in a physics simulation of brains, you would have an emulation of that process, right? The way by which neurons make new connections to one another. So it's like there's a lot of complex challenges associated with trying to do a brain upload.
No, absolutely. And so this is sort of like the whole idea of digital immortality that were initially, although we can prolong the life of the life expectancy or the life of our bodies with exchanging the maybe organs with something else, with like technology, but like the brain is the one thing that's limiting us so much. And here the whole digital immortality that we mentioned, that Richard just mentioned, there's a great series called Upload that sort of discusses this idea, but in a way it's a pro and again also an against argument, right? Because it's the whole concept is that when you have super corporations basically creating, as they call, heaven for you, like a life after death, but with ads, right? Type of thing, right? And if you can't pay, you get frozen, right? Like a basically computer gets frozen, shut down for the time being until the next month of payment or type of thing.
Yeah, so your consciousness is basically owned by it's the ultimate data ownership problem, right? your entire self is
owned by a company,
owned by
it's
by some Amazon or Facebook or Google
exactly.
entity.
And which and the whole idea as well of we mentioned this before by Elon Musk, like Neuralink type of thing, like with ads being played in your sleep type of thing. Like, so there are pros of like the whole idea of like using technology of expanding your life expectancy, but at the same time we have, with the way the life is and our world is at the moment, it's not the most appealing, I would say, way to do it. A temporarily fixed to it could go really, really wrong.
Yeah, so to mention a couple of other pieces of media that are interesting in dealing with that space of like uploaded minds. One of the kind of fun, optimistic ones that I really enjoy is the Bobbyverse books by Dennis E. Taylor. These will all be in the show notes. And then there's kind of on the darker and more like hard sci-fi exploration of the nature of consciousness and kind of philosophical aspects to it is Permutation City by Greg Egan. Right. If you're interested in philosophy of consciousness, like definitely read that book. That is a kind of a mind. That's just read Permutation City. It's great. And then there's a weird book that kind of sits somewhere between, I suppose you might call it speculative non-fiction, because it kind of has the structure almost of like someone who did the world building exercise for a setting, or a whole bunch of possible world building exercises for a setting in which there are emulated consciousnesses, but never did any of the like narrative work around that. It's by Robin Hanson, the economist. It's called Age of M, or M for emulation, EM, and that explores kind of the like really detailed real world consequences for what the economy would look like, and like everything from like power infrastructure to all kinds of weird stuff about what it would look like if we could relatively computationally inexpensively run emulations of human brains. But a lot of that also gets into this whole question of like identity and what does it mean to have multiple copies of your consciousness running, or what happens if like in Permutation City one of the characters has a copy of his consciousness that he's trying to figure out like to what degree can I screw around with how this is computed for it to still have like a sense of continuity of consciousness, right? Well, what happens if I run a scenario out of order? So I execute a whole scenario, but I do it like in parallel and back to front, right? Or so you know, I do all of the operations backwards. Does the person in the simulation still have the sense of continuity of consciousness resulting from these out of order operations? So you get into those kind of deeper questions of what does it mean, and then like in Robin Hanson's Age of M, you have all these questions about like, okay, what happens if I'm about the rights of these consciousnesses, and the economics of that, right? So if I have specialist skills, could I just like spawn a whole bunch of copies of myself that can go and work on these projects, and then like do they what should their rights be? Like, can I just like, you know, create a copy of me that I'm expecting to go work you know, like there's a whole bunch of really interesting questions around how that would work.
So there are certain sort of philosophical questions, you know, the whole idea of like, you know, which become relevant at that point because we're related to use of technology, you know, like the whole idea of, you know, teleportation and your clone doesn't die because, for example, if that's how it works, you know, you have to close to who's the real you type of thing, you know, the whole idea, the ship of Theseus type of thing as well, you know.
I mean, the ship of Theseus one is almost more relevant.
Yeah, that's why I was going to say that it's more relevant to the biological function. Like, you know, when become biologic immortal, like who are we truly, right? But in the same time, it sort of explains like, you know, like it's you continue maintaining like the you, right? But the change is part of who we are, so. It's not really, but for me, immortality, like for the social economic aspect, right? I'm a bit worried about it because, you know, the idea of, you know, having immortality being achieved, the first people who would have it, we know who they are, the richest of the richest, and the idea of like this whole idea of having the same people in power is there has to be a continuity of change, right? That, as you mentioned, like there has to be a change. It can't be just biological. It has to be social, right, as well.
Yes, yeah, and that's it's definitely a common argument that is kind of raised against the warriors. So what about the immortal dictator, right? What if Hitler or Stalin or Kim Jong Un were just able to live forever and continue to accumulate resources and deprive the population from it, right? And yeah, that's a problem, but it is one that I think is not like commensurate with having people die. Right? So the main thing that whenever people kind of make arguments against the idea of mortality is I like to start by just like let's invert the frame for a second and ask the question from the perspective of the world in which nobody like dies of intrinsic causes, right? Or even just the world in which everyone has a fixed rate of risk of death, right? We've attained negligible senescence, and everyone will more or less continue going on living unencumbered. Unless they're hit by a bus or analogous like super rare thing happens to them. right? In that world, do you solve the problem of there are some bad dictators by introducing a process wherein everybody slowly develops an increasing number of horrendous diseases, gradually falls apart, and just sort of, deteriorates and dies, right? It seems like this is a completely disproportionate response to the problem, right? If you propose that as a solution to getting rid of the dictators, people would probably not entertain it for very long because it sounds like a terrible idea. Yeah.
But it's funny because it's basically like it's a reverse of the issue we are trying to address here, right?
Yeah, it's like aging as a solution to basically any problem. I'm like, okay, your solution has to be worse than everybody dying. Also, your problem has to be worse than everybody dying for aging to be the solution to your problem. It's like it doesn't really stack up.
Yeah,
see what I mean.
see your point. But but still,
everybody dying horribly, I might add, in a protracted, drawn-out process.
But even then, like the whole idea of the cycle, because if there's going to be there has to be some, the effect of immortality would have significant effects on the society, right?
Oh yeah. I'm absolutely not saying that we would have to undergo some substantial sociological changes to transition to a world where our current institutions were capable of they're not configured for dealing with immortality, right? Even just like the institutions of retirement and kind of a three-phase life of education, work, and retirement, right? That's not compatible with a world.
Yeah, that would have to go out the window as well. Yeah, I mean, like that, and here I also wrote like pensions and retirement. The whole idea of like how does it affect the structure for things like retirement funding activities? This whole idea of like the way we are functioning now as a society would have to really change.
Yeah, we'd have to completely revisit that. I mean, one of the things that we might transition to is, I mean, it's already the case that our world is changing sufficiently regularly that you no longer train for a single career and pursue that career in exactly the same form for the rest of your life, right? That's kind of for the longest time that was the pattern, right? You apprenticed as a stonemason and then you were a stonemason until you died, or could no longer handle a chisel, right? And even up till more recently, you worked for a big company or whatever for your entire career. But now, like, increasingly the duration of a given career is in a particular domain is shorter and shorter. And people will have difficulty with like the retraining interval, right? So the thing that you would do in a society where people lived in they did not have deterioration with age would be kind of cyclical, right? You'd have periods of retirement and periods of retraining between your periods of work, right? You might work for a few years in a particular domain after having trained for a few years to specialize in that domain, and then take a few years off to relax before you restart your retraining cycle again with the resources you accumulated. Why not just rinse and repeat indefinitely, right? That sounds fun
but
to me.
this is why we come with the whole idea of philosophical aspects of the immortality, right? Because it's this is William's argument, right? The exhaustion of desires, right? Like you set yourself a end point, right? Like sort of like a goal. And be like, okay, you fulfill that goal, and then what's next? You make another goal, right? But like, at some point, like, what if you reach a fulfillment of the categorical desire, right? It would lead to like not wanting to live anymore, right? That type of thing. Like, if imagine like doing like learning all the instruments, languages, whatever, which obviously is not possible, like unless we are.
But so part of my quibble with this whole thing is like there is not a finitude of available experiences.
That's the
right?
thing with this,
as more people exist, they generate more stuff, right? New books that are good and interesting that I want to read are being written at a rate that is faster at which I can read books now, right? Right? This is already a problem of like there being way too much stuff that I could possibly do, and that growing at a rate that is greater than I could ever actually keep up with it. So like my like degree of concern of running out of interesting things to do and stuff that will keep me like engaged in life is like extremely low.
No, absolutely.
I can't envisage what it would be like to run out of new experiences, right? There's so many of them.
No, absolutely. I also agree with this this counter argument to the whole Williams argument, because like as you said, like at the moment we're working on something, but like the reality is that we can continue learning new things. There's gonna be always the development of something new that we can always grasp and try to achieve certain expertise in, and then by the time we do it, like there's more and more, right? The only problem I have is like the physical limitation of our brain capable of even remembering all of that.
Yes, yeah. I mean that's like, I mean, clearly a component of like a transhumanist future is one in which we make some improvements to to our brain, right? Because I mean, at least initially, I wouldn't object to a situation where I end up a little bit on repeat because I can't retain all the stuff that I've learned, so it goes away, right? That's the situation we find ourselves in now, right? We have to refresh our skills and our knowledge about a given area when we're doing something specialist in it, right? Because we don't have perfect retention. So like that process stretched indefinitely, but with just, you still get in the way that you do now, a kind of more abstracted intuition tuning impression of the subject material that you covered, even if you've forgotten its specifics, would still be valuable to me. But a part of the value of pursuing an extended life is the capacity to pursue extended capability, right? It's when I was like, I don't know, how old must I have been? Maybe like 13 or something. But when I first formed an interest in like longevity research, my reason for it was throwing up my hands and like I do not have enough time in my lifetime to figure out what exactly the hell's going on here, right? Like to answer the big questions, and so like the first step on that ladder is, okay, fix the immortality problem, because that way you have enough time to fix all the other things and figure stuff out. And one of those other things is the imperfection of human memory and the limits to the capacity of individual growth, right? They are merely challenges with which immortality or extended lifespan grants you the opportunity to work on solving if you have the available time.
No, absolutely. So, but in the same time, this whole idea of immortality also, there's an emotional cost, right, related to it, right? Now we were talking about only like the experiences, right? The whole idea of experiences, but at the same time, like there's also an argument of the emotional cost, whereas like the question, could you live continue living if your loved ones have perished, right? Type of thing.
I mean, this problem is relevant in the situation where like you as an individual are granted immortality, right? But I don't really see it as being as much of a problem if everyone has extended lifespans, because like if you're living indefinitely, then like, and so is everyone else, then you don't have this
absolutely
problem
no absolutely
of outliving your loved
absolutely
ones.
This is where we talk about the whole population is having prolonged lifespan, but then like if initially I assume like the initial stages would be the limited, right? There's certain steps to initially the idea, if we do reach that level of technological marvel that we are capable of introducing biological immortality, it's the fact that initially there'll be just a limited number of people, right? And the limited number of people is just the whole emotional cost related to it. But then again, as I said earlier in my argument, it's probably the rich people coming in, and those most of them are the ultra rich are like sociopaths and psychopaths. So it's probably they wouldn't care.
Mm-hmm. Yeah, I can certainly see that being like a strong reason why many people wouldn't want to live indefinitely if they couldn't bring the people they care about along with them on that ride, as it were. Right? The losing people is the worst, and extremely. It sticks with
absolutely,
you.
Absolutely.
It remains a part of who you are, and that would amplifying that indefinitely, and that's been explored in many forms of fiction. I'm trying to think of. I think this comes up a bit in one of the Gulliver's Travel stories, where one of the characters continues to get old and definitely lives forever, but just gets more and more decrepit, but also loses the people around them who are not subject to this curse. And it's there's a lot of places where that concept has been explored. It comes up in the Bobverse universe a little bit. the people who are not in Bob's situation of being an uploaded consciousness are referred to him by him too sometimes as ephemerals, right? Because they appear ephemeral to him. Which is which wears on him because he's losing people.
It's hard on people, but if we did reach a whole population wide immortality, I think this is a good point to mention the favorite.
Actually, one more little point on that.
Go on.
That last subject. So there's a book that I want to mention now because I didn't put it in the notes. I Am a Strange Loop by Douglas Hofstadter talks a little bit about the. It's kind of a book about consciousness, about the nature of consciousness. A strange loop is kind of one of the phrases that describes consciousness in Hofstadter's thinking about consciousness. But one of the core themes in that book is kind of the degree to which we keep people who are dead alive by emulating their consciousnesses in our own, right? That's the kind of the he lost his wife, and he talks quite a lot about that loss and how it affected him and his thinking about consciousness in that book. It's quite a beautiful book, actually. But in the context of and this notion of an immortal consciousness, like if in any degree we keep people alive by emulating the patterns of their thoughts in our own, which is a thoroughly scientifically debatable and a questionable concept, but if there is any truth in that concept, then being immortal and carrying the image of the way that a loved one's mind works forward with you into immortality would be something that I think would be have a certain poetic beauty to it, even if there is a great pain of loss.
Yes, absolutely. Yeah, I think we pretty much covered everything in here. Except for the one thing that you haven't mentioned, but we have sort of discussed this, like the whole idea of wanting to continue to the experiences, right? This was, I think this is greatly influenced by the book that we both love.
Yes, I should mention Harry Potter and the Methods of Rationality. Because I think the Chapter 39, the latter half of Chapter 39 of Harry Potter and the Methods of Rationality more or less perfectly sums up my attitudes towards immortality. We've mentioned HPMOR quite a lot before on the podcast, so I imagine our audience is familiar with it. But like if you haven't read it and you have no intention of reading it, but you listen to this episode, then the latter half of Chapter 39 is well worth a read. It encapsulates many of my thoughts.
No, honestly, it's great to have. But at the same time, I feel like at the end that I don't know. It's in the same time, I feel like it depends on like the circumstances that we end up with, right? The whole idea of immortality, where do we begin with the whole, the whole society being immortal, or whether it's initially the individuals just being immortal? And for me, I don't know that it depends on like how we address the problems that we raised together today. And overall, I do agree with certain point that I would like to experience more things. As you said, there are so many books and things that I would like to experience still. But at the same time, I'm still for the whole idea of the continuity of the cycle, right? The whole idea that the use of resources, would be quite substantial compared to obviously we're talking about to reach the all biological immortality would require a long time of still research and technological advances, but at the same time, like if
actually so one of the concepts that we should probably bring up at some point is longevity escape velocity, right? Which is a notion popularized by Aubrey de Grey, wherein like the technology to extend life continues to be developed the longer you're alive, right? So in fact, your lifespan on average for the past several hundred years has been increasing by about 6 hours a day. So like the longer you stay alive, the longer you are likely to continue to stay alive because of developments in technologies which increase the likelihood of you staying alive, right? So if we reach the longevity escape velocity, right, it's the point at which the rate of increase in our life extension technologies exceeds the pace at which we're aging and dying, and which people being born today, and maybe even some of the people already born, may be in the appropriate part of that curve to have reached longevity escape velocity, and will continue to live more or less indefinitely because the technology is improving at a rate that will catch them in that before they die. They'll have reached the escape velocity.
No, absolutely. I guess what's your? I mean, we know, but like, could you summarize your thoughts, Richard, about this whole idea, about being immortal, like the whole about immortality?
I mean, I suppose it's. I mean, for me, it's difficult to summarize it because I don't really understand the concept of like not what. Like, I mean, there's a quote from that Harry Potter Methods of Rationality chapter I mentioned. Right, it's like, I want to live one more day. Tomorrow I will still want to live one more day. Therefore, I want to live forever. It's proof by induction on the positive integers. Right. It's like, if you don't want to die, it means you don't want to live. It means you want to live forever, right? If you don't want to live forever, it means you ultimately want to die, right? And like, I don't particularly see the appeal of no longer existing, right? There's so much that one could potentially do in life, and I don't see why you would like turn that off unless your life was like particularly unbearable. That's I don't get it, right? It's I say it's hard to articulate.
I understand. I think it sort of explains what I partially agree, but at the same time, like it depends on the circumstances we are surrounded, like, and you just said it, right? So, but I guess this pretty much summarizes everything what we talked today about, right? We wanted to talk about.
I think so. We covered. We took quite a while doing it. It's like 3 hours now.
3 hours now.
But we covered a lot of ground.
But
I mean, there's still a huge amount more to this subject than we could cover in this conversation in 3 hours. There's a fairly extensive reference list of additional places to go that I wrote down that we'll be putting in with the show notes. And there's a lot of really interesting fiction that covers related subjects. I'm almost done with Surface Detail, an Ian M. Banks novel in the Culture series, which explores some of the questions that we were just asking about sort of simulated consciousnesses and whether or not you might want to live if your existence was unbearable, without giving too much away. There's a lot of other places to point to. We've scratched the surface here, and I had another conversation on the Bayesian Conspiracies podcast some time back now, when I talked about some of the same stuff we covered here, went in a couple of minor different directions, if you're looking for more on that subject. And then there's a bunch of other podcasts, including one from David Sinclair and his book Longevity, and a bunch of other popular books on longevity science are going to be linked to the description. So there's you've put up with 3 hours of us talking about it. So,
but it's only the beginning.
You may want
it's
more.
only the beginning. Yeah,
exactly.
But I guess that's it. Thank you very much, everyone.
Yes, thank you very much for listening.
We are Xenothesis. You can find this podcast on xenothesis.com. Today I was the host. Yes, that's the word I was looking for. Yeah, I know. It's honestly, okay. I think our brains are fried at this moment. But yes, thank you very much, everyone. I was Mike Glinka.
And I was Richard Acton.
Goodbye.